Marsilia minuta. We all know that Bengali name is Susunichak, and that is the ampelop tree, that is the young fronds at sarsenet varnation that is sold in the market, and that is Hyathiya. And if we just take another plant that is Metuxia tutriopteris, that is known as ostrich fern, and its sarsenet coiling vernation is looks like the violin, and for that reason it is known as fiddlehead ferns. And the young, tender, tidally furled, new growth shoots of fern family plant, and usually this is known as common name is ostrich fern, and it is Metuxia tutriopteris. So it is used as salads in some of the countries, and that is looks like that is the palatable dish is there, and you just add some butter, salt, pepper, and maybe some lemon. It is a good palatable dish. So Metrix tuchiopteris is also used for this purpose, and this is known as fiddlehead ferns. And if we go to the competitive nature of this particular plant, that is the how that is good for health, we find that is unique in taste and carry just 34 calorie per 100 grams. So at the same time, more 100 grams only, that is 34 calories, that is higher than other angiospermic plants or commonly used vegetables, and consist of health benefiting antioxidants. We know that antioxidants is more, and vitamins and omega 3 and omega 6 essential fatty acids are there. And another is that fresh fronds are very high in antioxidant like vitamin A, carotene, and 100 grams of fertilizers hold 3,617 international unit, or 120% of the recommended daily requirements of vitamin A. And they are excellent source of very natural polyphenols, flavonoid compounds such as alpha and beta carotenes. And their sweet taste, this taste is sweet due to presence of vitamin C. 100 grams of fresh prawns contains 26.6 milligrams, or 44% of the daily required levels. And vitamin C is moderately potential water-soluble antioxidant, and flavonoid compounds like carotene. It helps scavenge harmful free radicals and offer protection from cancers, inflammations, and viral cough and cold. So that is the natural antioxidant properties, and also rich in vitamin C. And it is also good source of minerals and electrolytes, especially potassium, iron, manganese, and copper. And 100 grams of fresh sprouts carry 370 milligrams of 7% of daily required levels of potassium. We know that potassium is good for health, and potassium is a good electrolyte, and it is a friendly minerals for our heart, and it reduces blood pressure, heart rate by countering the sodium effects. Further, they contain small to moderate levels of some of the valuable beta complex. So, as for example, we just go to the comparative nature or why the pituitary funds are so important as a food, and if we just use these vegetables. And there is a little pteridophytes is not only the neglected group of this plant, that's from the because that particular plant is not so much exploited in the in our in this particular seminar, that when we concentrate on the food security, the there may be the main staple food is there, supply is plenty, we just supply any man or the human popular civilization in the jungle or the tribal elaka, they just collect their staple food, but they are lack in nutrients. So that is in this modern scenario, that is in this condition when the child and the adult peoples are less in immunity, and especially in this pandemic situations also, they are less in immunity systems or deficiency of many micronutrient and macronutrient. So if we just focus on this particular group of the pteridophytes, and we exploit or concentrate on this, and the peoples only use these plants by virtue of their knowledge from generation after generation, and but not totally focused on this particular group. That group is especially neglected group, and for this reason we just go to the concluding that it is important to identify the potential sources, which material or which minerals are there, and it may be used as alternative food, especially in that region where the human civilization is not reached, and where the human populations, especially child or adult, are not totally conscious about their immunity, about their deficiency, about their nutrition. So that is known as highly deficient region. And in recent years, various lesser known plants are being evaluated for their nutritional and medicinal properties. But there are still a large group of species that are commonly eaten and sold at local market whose nutritional potential has not been studied. They are only sold in local market. They are not estimated properly, and they are not in this modern scenario that in the age of this advertisement, in the age of this social media, they not get so much populated to human civilization. And among the wild plants, edible ferns can play an important role as they are often included in the daily diet by the populations of the eastern Himalayas. And we should take some responsibility to ensure a healthy natural habitat, to develop a protocol for sustainable utilization of wild funds through value addition, preservation, packaging, processing, and marketing. And their harvesting must be focused. Their harvesting should be regular, and their harvesting should be not over harvesting to destroy the species, because so many plants are even now not described, so many plants are even now evaluated phytochemically. So harvesting requires instant monitoring and management of local strategies by taking into account the involvement of local communities. And modern farming techniques familiar to the locals can be developed to fully meet the demands of the underutilized plants. Like the underutilized means that are not so much utilized. That is not so much utilized throughout the all over India, all over the population, or all over the country, and thus developing these wild species into cultivated ones. They must be cultivated as we cultivated some flowering plants, as we cultivated some ornamental plants, and as an alternate cultivation of the by means of crop rotations. So we must gather some knowledge and we must exploit this particular group of plants. And at the same time, making ferns and fernalies a compatible vegetable variety. So these are the references here. And so that is the jungle of ferns. And so that is my complete end of this lecture. Thank you.
Okay.
Hello.
Thank you, Dr. Vandari.
Yes.
Thank you for your nice talk on wild edible ferns, which is very important nowadays, that we have to find out much more species, wild species through traditional knowledge, and that should be channelized in the food habit, agriculture, and also in the change of food habit of human beings. Okay, thank you for such a nice talk. And the list of
Okay.
pterophytes of the group
Thank you.
Lycopodiophyta, and their uses and their nutraceutical values, that in every point you have enlightened us. Thank you very much. So now we have Dr. Sarup Kumar Meena. Good afternoon, sir. Are you there?
Yeah. Good afternoon, sir.
Good afternoon. Good afternoon. So now we have with us Dr. Ram Sarup Meena from Banaras Hindu University, and his title of talk is Soil Organic Carbon Management for Food, Nutritional and Environmental Security. So before start his talk, I have a small introduction that I have to read in front of you. So formally, actually introduce him to you. Dr. Ram Sarup Meena is born in a farmer family and working as assistant professor in Department of Agronomy, Institute of Agricultural Science, Banaras Hindu University, Varanasi. Mr. Meena has secured first division in all the classes with triple net junior research fellowship and senior research fellowship from the Indian Council of Agricultural Research (ICAR) and RGNF awardee from the University Grant Commission, Government of India. Dr. Meena has been awarded Raman Research Fellowship by the Ministry of Human Resource Development Government of India. He has completed his postdoctoral research on soil carbon sequestration, underworld food, uh, underworld food prize 2020, and Nobel in contribution 2007 laureate Padam Shri Professor Ratan Lal, distinguished scientist and directors, Carbon Management and Sequestration Center, the Ohio State University, USA. He is working on soil sustainability, crop productivity, and resource use efficiency under the current climatic era. Dr. Meena has supervised 25 postgraduate and 6 PhD students, and he has more than 10 years of research and teaching experience at the undergraduate and postgraduate PhD level. He is working on the four extremely funded running projects from IOE, BHU, DST, MHRD, ICR, and involved in many academic and administrative activities going on at the institute or university level. He has the right to one patent on low-cost biochar preparation. Dr. Meena has published more than 80 international research and review papers in peer-reviewed reputed journals, up to 9.3 impact factor with H index 45, and citation is more than 5,250. He has published 4 books at the national level and another 19 books sponsored by Springer, Elsevier, etc., at the international level, and contributed to the books with 15 chapters at national and 50 at the international level. He has worked as an expert in the National Council of Educational Research and Training, MHRD Government of India, to develop the 2 books for school education. Dr. Meena has been awarded several awards, namely Young Scientist, Best Article, Young Faculty, Global Research, Excellence in Research, Honorable Faculty Award, etc. Dr. Meena is a member of 9 reputed national and international societies and is working as a General Secretary, Editor, and member of the editorial board in 12 national and international peer-reviewed reputed journals and attended several national and international conferences in country and abroad. Dr. Meena is contributed to the agricultural extension activities in the villages, active as an associate coordinator in pieces of training, meetings, workshops, and farmers fairs. So this is a very little introduction of Dr. Meena. Now, on behalf of Department of Botany and the chairman of the of this conference, so I welcome you in this virtual platform and would like to request you to present your lecture. Over to you, sir.
Thank you, respected sir. Manoranjan Chaudhary ji, sir, you talked me about a lot. I think I am not deserve
No,
for a
sir.
long introduction because just I
That
am
you
an
deserve.
assistant professor, sir. So, first of all, I would like to say thank to Chief Patron, Honorable Vice Chancellor Subresh Bhattacharya ji, respected Dean Subhash Chandra Roy ji, and I also thankful to you, sir, just to introduce me, and as well as organizing secretaries Piyush Mathur ji and Dr. Surenendu Roy ji. He contacted to me to deliver a lecture, and as well as I also thankful to members Professor Saha, Professor Sen, Dr. Bhandari, Dr. Mandal, and some other members, Dr. Bhattacharya ji and Dr. Hussain. Just I think you assigned me half an hour, but I requested to you, sir, kindly count my half an hour from I'm just starting. So because almost 12 minutes are gone from my half an hour, so I would like to share my slides, and if you will permit me, sir, I will start. Sir, my slide is visible, sir.
Yes, yes.
Yes, sir.
Dr.
So,
Sharup, visible.
Okay, thank you,
You
sir.
go ahead.
So
Visibilities.
my topic is because your title is Advances for Plant Science for Food Diversity and Nutritional Security. But I'm completely focused on the food and nutritional security, but as well as I will cover the environmental security there. So I will discuss today related to some aspect of the agriculture and soil organic carbon, because two type of carbon in the soil, active and passive, organic or inorganic. So I am completely focused on the soil organic carbon. So what is the relation of the soil organic carbon? Resource use efficiency, crop productivity, grain quality, food security, nutrition security, environmental security, economic security, soil health restoration, and human health, because soil health and human health completely related to each other. So all these aspects I will cover in my talk. So yeah, now I start agricultural production after green revolution and its impact. Salute to that scientist, Dr. Norman Borlaug, Nobel laureate. He started the production from 1000 to 5000 kilogram per hectare. Or especially green revolution with the varieties of the wheat and rice. Initially, two crops are focused and increase the food grain production like that: wheat 208%, rice 109%, and maize 157%. That is the situation of India from green revelations to 2019. It is right now. It is more upper side. At that time, per capita availability of the rice is 58 kg. Even population was 350 million. Right now, we are getting around 60 kg per year per capita. Even population is very high right now. So parallelly, wheat yield also increased. So we are getting from 24 kg to 70 kg right now. It means that is the result of the green revolution. The boost up the yield, the so many aspects play important role: hybrid seed, irrigation, fertilizer, pesticide, and mechanicals. But in the other behind the curtain story, some negative issue is there. Or I will focus right now on the chemical issue and malnutrition issue, because we must be go to the food security or nutrition security. But I think in my personal opinion, we are not suffering from the food security right now. We are suffering from the nutritional security, and some climatic aspect is there, because even in India, if we can see the example in FCI godowns, or millions ton of the food grain is spoiled. In the, we can say, we don't have particular facility to store the grain, or even government is purchasing under the force of voting. So many political issues there. So we must be focused on the nutritional security. But due to the chemical, we are continuously increase the chemical consumption in India. Especially, we are contributing 76% insecticide, whatever agrochemicals we are using. So result is there. One example I am taking from the Punjab. Punjab right now is a hot spot of the cancer. But we are running to go to the cancer, different type of, we can say, treatment of the cancer. But we are not thinking what is the, we can say, root cause of the cancer. One, even I was in PhD, one train is running. I was in PhD in Bikaner. So one train is running between the Bhatinda, Punjab to Bikaner. So name of that train, if you will go to the inquiry, we will ask the when cancer train will come. So the window person immediately, we can say, clear the time. So result is there. The number of patients is coming from Punjab to another states, because we have Punjab and some other states, their consumption is very high about the agrochemicals. So that is the reason. Or so many type of pollution, we can say soil, water, air pollution, food, even plant, soil to plate, everything is contaminated with the pesticides. So we must be think about the agrochemicals, different type of agrochemicals. They have effect on the soil as well as environment. Or like that, recently government of India banned the 20 chemicals, because they are coming to the plate from field to, or we can say, soil to plates. So that is very important. We go to the solution about the whatever the health issue is there. Even one example, our children are getting stunted growth, or even in the index we are behind of the Pakistan. Because Pakistan have good position about the nutritional security than us. Relation of the soil sickness and human health completely, especially we just in applying the NPK, we are ignoring the some other aspects. That's why reason so many deficiency like that. 49% of the soils in India is potentially deficient in zinc and 12% of the iron. So women are especially suffering from the iron deficiency, or as well as zinc deficiency. Every health issue affect the zinc deficiency. So due to that type of deficiency, just we are applying NPK, the pharmaceutical industries business are carry on to the upper side. Many millions of rupees we are investing to the supplement. We can say, just if you will go to the doctor, they will give the 1 or 2 tablet for the particular disease, but parallelly they will give the supplementary tablet and some we can say supplementary powder like that. So many things there. So that industry is flourish completely because we are not getting the nutritional food. So recently, my one article published, it was the free or invited article, and it was we can say awarded with the best award with about 1.5 lakh rupees way of charged, and that is the case prize. So we completed, and that within a year, I think we got about 175 citation from that. That is totally depend on the how is the soil health is affecting how impact us agrochemicals on soil microbiota, because soil microbiota is a key of the soil and how it is affecting from the agrochemicals. But next challenge is there. Soil is affecting, or soil is sick, or food is sick. Ultimately we will be sick. It means we are not getting proper food. Parallelly, next challenge is climate change. But climate, we must be think about the climate change. So everybody is not, I think that seminar is also covering a good aspect. I've gone through that, we can say things. So I will not discuss about the what is the climate change, but ultimately main pillar of the climate change or greenhouse gas emission is carbon dioxide. So ultimately we must be focused to the carbon dioxide, because just we are emitting the carbon dioxide to the atmosphere, and that is, we can say, changing the so many things is happening in the climate or with the climate or atmosphere. So we must think majorly, we have need to think on the carbon dioxide. How can we reduce the carbon emission from the earth? So it is not a matter about a country. It is, we can say, responsibility to anybody who is living on the earth. So we can say a single unit is also responsible to reduce the result is that due to the greenhouse gas emission, climate change. Over there, that is the picture from the Kashmir. Most of the time snowfall is there, but during the period of the snowfall, we are getting rain, or even rain is up to the flooding condition. But somewhere we are getting the drought. So something is happening due to the climate. Some is severely drought, some is severely flood, like that. Everything is happening with the. But being an agriculturist, I will focus on the agriculture. So what is the contribution of the agriculture? Agriculture is contributing that 28% to the climate. And from carbon side, this is that article recently came in the Advances in Soil Science. It's a world-renowned series of the soil science, and that sectors are contributing from the India. Parallelly, we are also playing a great, we can say, very important role to how can we check the emission, how much equivalent we are emitting from the agriculture side, but how can we reduce that one? It is very important, because every crop contributing to the CO2 emission. Parallelly, now shortly days will come. That picture, that type of picture, every day we will see on the news channels. So every plant have almost, as per thumb rule, 40 to 45 or 50% carbon. if we will burn the crop residues, it will emit to the atmosphere in the form of CO2, or parallelly from the field, we are emitting millions ton of the nutrients from the soil in the form of nitrous oxide, sulfur, so many nutrient is there. Even we are giving the subsidy to the companies to produce the fertilizer, so many things is there, but we are not thinking how can we manage the things. Just we are blaming to the farmers, but it's my personal opinion. I think farmer is not, we can say, responsible for that, because our policy and planning is not perfect. Otherwise, he's not ready to burn. He knows something about the value, but to get the next crop, he's, we can say, due to the shortage of the time, he have need to burn the crop residues. So ultimately, if we will stop the crop residues, that type of problem we can manage. Because due to the burning, so many issues are linked to each other. Residues burning, adverse impact on soil quality, air pollution, greenhouse gas emission losses, millions ton of nutrient we are losing, soil biodiversity, because macro, microorganisms, macro nutrients, so many things. Parallelly, number of diseases are coming to the society due to the burning and air pollution. And depletion of the soil organic carbon pool. Soil organic carbon pool is a key. We can say like that 100 locks is there in the soil, but one key is there. It is a soil organic carbon pool. So that's why reason, just I coded in my last slide, every solution with the soil organic carbon pool. But that is the question, how can we manage? Because everything in ecosystem services, soil organic carbon play an important role. He's the energy of the microorganism. He is, we can say, help to mineralize the micronutrient to the plants. Recently, we did the study on that aspect, that microbial, we can say, diversity and nutrient dynamics is also affecting due to the burning of the crop residues. Parallelly, we are also blaming to the farmers. It's a picture from the Delhi, or most of the burning area is NCR region. So during that period, increase the air pollution. Parallelly, but we are just blaming to the farmers only. But I think so many things there with the not a only single crop residues burning. That picture is showing, if lockdown is there, it was the picture from the lockdown, that is the normal life. Ultimately, it's a picture of the air pollution. It means industry is also stopped, motor vehicle and automobile industry is also, and vehicle is also stopped during that period. That is the reason. So what is the result of that? That is the another business is started. That is a picture from the and coded from the Delhi. A new business is started. 50 minutes breathing session. Amount is around 500 rupees. It is because the, we can say, completely air pollution is there. It means CO2 level is very high. Other than CO2, so many global warming gases is there. So ultimately, oxygen bar is opened like that. So many bar is there in the country, but we have still we come to the door to open the we can say oxygen bar. So maybe it is right now in Delhi. In the future, maybe it will be in your city. It will be maybe in my city also. So we must think parallelly about that one. So that is the indication of the nature. So we are suffering of the shortage of the oxygen. So just we have need to think about that. Otherwise, future will be very dangerous, under the dangerous situation. Because not a just matter about the agriculture sector, parallelly main players from the industrial sector, but nobody is focusing on the industrial sector. Just due to the political issues, just we are promoting the industries. Yes, there is the need to economic of the country, but we have must need to think about the pollution. So just we are blaming to the farmers. Being agriculturist, I am not, we can say, happy that type of decision. Otherwise, farmer have every solution. So I will show in the next slide how farmer have every solution. So it is very important that if that type of situation will be continuing in the coming era, the temperature will be rise, or even some evidence showing that if 1 degree temperature will be increased, Like that, I will give the example of the wheat. If 1 degree temperature will increase during the maturity of the wheat, 4 quintal will be yield will be reduced. That is the, we can say, steady. But parallelly, so many things is also happening. But industries only have emission of the CO2. But in agriculture and forest, agroforestry system have solution. We are capturing the CO2 from the atmosphere in form of biomass, because we all are from the scientific field, we know very well during the photosynthesis plant have need to CO2 and it will convert from the biomass. And just I quoted one example, every plant have biomass 40 to 50%. So if that biomass is captured or CO2 is captured from the atmosphere, we must be have duty to stay in at the soil and upper biosphere. Even in agriculture, we are continuously have erosion aspect. That's why reason the agriculture soil carbon is also emitting to the atmosphere in form of CO2. So we must be think about that, how that CO2 we will stop from the to emission from the agriculture, because that CO2 is also carbon or soil organic carbon in the soil. So just I quoted the example, soil carbon is a key of the 100 locks in the soil. Every solution with the soil organic carbon, and soil organic carbon convert to the CO2 and emit to the atmosphere. So ultimately in Indian soil or any, we can say, productive soil, we have must need to about 1% soil carbon, but still we have 0.2 to 4.4% carbon. It means there is lot of scope to increase the soil carbon, or even not a matter of the increase the soil carbon, that is the matter to capture the atmospheric CO2. In technical terms, we can say soil carbon sequestration. So we have must need to think about that to restore the soil carbon. That is the big question. What is the solution of soil, human health, food, nutrition, or economic security, as well as for environmental management? My answer is soil organic carbon. But yeah, that is very important. How we can say soil organic carbon is a solution for the all aspects. Reason is that that program, Conference of the Parties, COP21, recently under that program, or Government of India constituted we can say body under the 14 Ministry of the Government of India. They are ready to work under that. So that agreement was in 2015. It was the COP21. So in that program, we have must need to restore or capture the organic, we can say CO2 from the atmosphere and put it in the soil. So if we will do like that, the CO2 level of the atmosphere will be, we can say, come to the initial phase within the 24 hours. I worked in that program with that, with that personality. He's, we can say, god of soil science. So, sir is just sir is introduced. Sir is got a Nobel Prize in IPCC with contribution, World Food Prize 2020, recently for Padam Shri from the government of India. So, but question is there, where is the soil carbon? Where does soil carbon comes from? Soil carbon comes from the atmosphere in form of CO2, but where it does? It is also is there. Because the emission of the from soil, agriculture bodies, the CO2 is there. Not only from the agriculture field, from the every aspect, automobile industry, so many greenhouse gases emission sector is there, industrial sector, but agriculture is also contributing in that one. But from the all soil, every industry is not able to capture the carbon. But agriculture have capability and soil have capability to hold it up. So how can it will be possible? That is the structure of the soil. 20% water, 20% air. It will fluctuate according to the water and air is available in the soil, and that 45% inorganic material, 5%. Every game in the soil with the 5%. Or especially that is where the solution of top 15 up to 30, 30 to 15. That is depend on the which type of soil is there. But my question is, their solution is here of the top layer of the soil. That soil is enriched with the atmospheric CO2 in form of the biomass, insert the any type of crop residues, planted the tree have also 50% biomass. So if we will promote that type of activity, so atmospheric CO2 will be captured and it will convert to the total organic carbon. So in the soil, every solution with that one, alchemical property, biological property, physical property. So that is the key of that type of. That article recently we can in last year published what type of long-term impact of that one. So it is very important. It will increase the soil texture, aggregate stability, so many aspects of the soil. That is the issue. So we must think about that type of activity. So just my request to all authorities, don't go to rule for the farmers. Just we have need to promote the farmers with that type of, maybe think about that. Like that carbon tax. Just we are putting so many type of taxes. You and we are bearing so many taxes. But my request is here, we must be put the carbon tax to those are emitting the carbon or CO2 to the atmosphere, or especially for the industries. But we are not focusing on that aspect. Just we are focusing on that type of aspect, short-term aspect. We must be think and get the carbon tax from the industries, and we put it as an incentive to the farmers. And we must be promote the farmers. So because time is shortage, I will skip some slide. Soil carbon restoration. So in India, we have need that type of policies. Soil organic carbon and its role in soil, environmental, and so many, I already discussed policy. We have much need to reform the agricultural policy education about the soil organic carbon. We must be focused on the soil organic carbon and promote it to restore the soil. What is the organic farming? Just we are trying to putting the organic carbon in the soil, nothing more than this. So we have must need to focus on the key element and key answer. How can we manage the everything? That type of situation is there. So support the some incentive finance, like that financial. We must need to promote the research on the carbon aspect. Just we are promoting the old model projects, millions of rupees we are investing on that. But I think it is not good, because it's my personal opinion, and that is documented with the advances in soil science. It's a world level series. So we have must need to think about that in Indian condition. On farm implementation, we recycle the whatever the agricultural waste or agrochemicals is there. So we try to replace the agrochemical with the agro waste. Motivation, payment for soil carbon incentives. Awareness. If sometimes we face the crime and we feel that is the crime, we come to the road. But we are not thinking about the environment. We are not thinking about the soil. Even we are investing the millions of rupees to the pharmaceutical industries to protect the environment. But we don't have any plan, or we can say key element solution about that. So I think it is very important. So sector-wise, it's a carbon sequestration. Parallelly, so if we will plant it alley cropping or we follow the agroforestry system, it will play the very important role, not a matter about the manage the soil health, atmospheric. It will also manage the nutritional security and food security. We planted, we can say between the fruit, we planted the crop. So we can say increase the income also, as well as manage all resources. So that will be the win-win situation. It means we capture the atmospheric soil, increase the soil organic matter. So ultimately we increase the food quality. Organic matter means just we are getting the organic food. So increase the soil quality, environmental quality, as well as increase the production or yield or get the good money. So that is another aspect of the soil health restoration. That's why reason topsoil. because I also coded in last slide that upper layer or topsoil is have every solution. Topsoil erosion save the our future. That's why reason we must be think about the topsoil. This book was the best book on the in the Springer in 2019 to 20 on that aspect. So we must need to manage the resource use efficiency. It means we manage the resource use efficiency. Carbon emission is also managed. Sometimes parallelly, even in India, we are wasting the food. From the one day wastage, some very small country, they have thousands or lakhs of the population, they feed throughout the year with the waste of the or 1 year food. So it like that, if we will produce the 1 kg of rice, 300 liter of water is there, number of labor charges is there, number of we can say input is there, agrochemical input is, soil engagement is there, so many things is there. So my point is here, please don't waste the food. If we will manage the waste of food, it will promote the food security, malnutrition security, nutritional security, environmental security, reduce the water footprint, reduce the carbon footprint, reduce the nitrogen footprint. Because like that we are applying the 1 kg nitrogen. So many story behind the production of the 1 kg nitrogen from industry to farmer field, or even some subsidized issue is there. So ultimately, we must think what we have aimed to birth on the earth. Just we are exploiting the natural resources. Just we have no need to think to the society. So ultimately, what we have footprint, it means what we are doing. We must be think about that. What just we are exploiting from the nature, soil, environment, or from the earth, and in the we can say another we are giving some replacement to the nature or planet. I think so that is the issue to think about that. Whatever serving to the society and nature, what is our balance for future generation? Just we are exploiting, we are cutting down the tree, we can say we polluting the soil, water and air also, but I think we are not thinking what we are giving the back to the nature. So I think definitely are we exploiting the nature? So we must be think to the answer, because the population during the 1800 to 155 million, but right now we are almost here. So resources are same, but continuously, drastically resources are decreasing. So we must need to think like that, because it is not our right to exploit the nature. We can say in one thought, I quoted, it's American thought in Hindi. हमें ये ग्रह हमारे पूर्वजों से just I want to explain the sustainability. Some persons are coded the definition of the sustainability. Whatever is there, we use it and save for the future generation. But who is the responsible? We already degraded that. So we must need to do something. Otherwise, I don't know what will happen in the future. Recently, we published that book in the elsewhere on that aspect. Parallelly, we are working on the organic matter. But I also have one question. We are running the millions of the rupees project to add the organic matter. But my question is there, no, I am not, we can say, satisfied that type of activity. We are applying in with the government is promoting that type. Yes, we must be do, but we parallelly think about that to increase the carbon stabilization. Just I coded here, because whatever we are applying in the organic compost form, it is again going to the environment. I completed that work. It was that 14-year-old, we can say, field in US, as well as we are applying the 25 megagram per hectare FIM, but nothing is, we can say, carbon is not stabilizing at the ground level or in soil. It is again going to the atmosphere with the oxidation. So now we have duty to think to stabilize the atmospheric carbon in soil, not just application. We must be think about the stabilization, because just because that is my conclusion. we are working for 1 to 5 year only. Right now, whatever we are working to the soil, atmosphere, agriculture, but according to my one SERB DST project, we are working for 1 to 5 year only. Parallelly, I also worked for 20 to 40 year in US. But I think if we will just like that, it's very simple. I don't know, because so many field person is there. You are getting my point is yes or no. I don't know, because whatever we are applying, it is again going to in CO2 form to the environment. But my question is here: we must be apply whatever we are applying soil. It will stay in the soil more than 200 years. So right now, our research, our government, our scientist is working here, and we are investing millions of rupees for that aspect. But I think we will do something for that one, because I worked in that on that aspect, and I also worked up to that 40 year. So my one project is also in pipeline with DST. So I am thinking we will stable the atmospheric CO2 for 200 years. So we will get double benefit. There no need to apply the fertilizer. It will increase the organic matter for long term sustainability, as well as we will capture the atmospheric CO2 in terms of the soil organic carbon. So we will capture it in the soil, capture the atmospheric carbon in the soil. So it is very important we think like that. Yes, recently that book is only book published around the globe on carbon stabilization. So the need to healthy soil for healthy world. So if we will increase that type of practices, we must need to follow to increase that type of activities. So our thrust area must be soil, human, environmental health management, relation management of efficient input or output, efficient economic utilization of the inputs, improve the farmer economic development index, agricultural sustainable intensification. We must be intensify the sustainable resources, eco-intensification, nutritional security and health index. So that type of work I covered in that one, or that is totally depend. That is my team, my PhD or master scholars, and this the primary Prime Minister adopted village extension activities. Thanks to all. I think I took much time. I extremely sorry for that, or yeah, I think I very completed my task in very short,
Yes, sir. Yes, sir. So,
because I was afraid about the time.
no, it's okay. It's okay, sir. It's okay.
So if anybody want to ask the question
Yes.
most well, I will be happy to give the answer for more or more questions.
So, anybody have any question? If you have any queries that you want to share with Dr. Meena, so he'll be happy to answer.
Because sir,
Sir,
I am
sir,
sorry
there
for
are.
that. I was so fast.
No, sir, sir, your lecture was very interesting. Actually, many of the students have joined through YouTube, so we are getting questions over YouTube. We will share. If you, with your kind permission, we can share your link to them so that they can visit your website and university site.
Why not, sir? You can share, but I have little bit, with the last slide, I put something important. So, but no issue, you can share it, because one my DST project in pipeline. Actually, it's a
Okay.
very big project. Like that, just I coded. It's a just we are working only for 1 to 5 year. But I also explore the soil. Only one lab on the soil carbon in US. I worked on that one for up to the 40 year. But my target is that we will stay that carbon to the more than 100 or 200 years. So it will be good for me. But if you want to share, it's your choice.
Thank you, sir. Thank
Okay,
you for the
okay,
very.
Dr. Meena, thank you very much for your such a nice informative talk on soil carbon sequestration, soil carbon management, air pollution, oxygen demand, increase of greenhouse gases, and their management, and lots of things you have shared with us. That are the real findings and the very alarming findings of your research. And obviously, the farmers are not responsible because that burning of those uh, plantlets is a temporary issues. Whether the industries are releasing every day
Yeah.
by day, without.
Actually,
Yes,
sir, I will interrupt. Actually, sir, we are just focusing to the farmers because in the November they must need to transplant the wheat.
yes,
But
yes,
our policy, I think I'm not. My one paper is recently came on the policies on soil organic carbon in the India. So if we will promote the farmer to give the incentive to increase
yes,
the
yes,
soil organic carbon, they will be not happy to burn that one because ultimately they want because farmer is doing farming because I am also a farmer son. So I know
definitely,
the status of the farmer.
yes,
If
yes,
they have good earning, so what is the need to burn it? Parallelly we are running the project, even government of India are running the millions of the rupees project. I am not blaming to the government or researcher system, but that is my conclusion. Whatever I did, my past, whatever my finding, I share to the scientific community. Whatever I have evidence, accordingly I am discussing to you because my mentor is a authority in that field. So I am so lucky. लिटिल बिट यू नो समटाइम एक कहावत है हमारे कभी कभी जैसे गुरु से इतना सीखता है चेला उस रास्ते पर चल पड़ते हैं तो सम वी एक्सप्लोर मोर so with the direction of my mentor I am running on that path.
yes, yes. Another thing is the sustainable development. This is very important things. So the destruction of forests and the other natural resources is a very faster manner. It is going on in very
Yeah.
faster manner. So how to stop this or how to slow this? This is our main aim of thinking. So it is a really alarming things that you have shared with us. So Dean sir, are you want to say something? No. Okay. Anyway, thank you, Dr. Bina. Thank you for sharing your valuable that outcomes, research outcomes with us. Thank you very
No,
much
I
for being
just
with us.
congratulate the Ramsharup Mina for his good presentations.
Yes, yes, yes.
Sir,
Really,
it's
I
your blessing.
delighted. I
Sir, I
delighted
want
because
blessings also.
because we are not the agricultural scientists, but from
Yes,
botany sides.
yes,
So we are learning the soil science, soil health. India government has given the soil health card for each of the
Yeah,
farmers so that we can improve the soil health.
yeah, yeah.
Without soil, we cannot harvest the more crop per unit area of the land.
Yes, sir. Yes, sir.
So, okay. Thank you,
okay. Thank
Ramsharup,
you.
Mina. Thank
Yeah,
you.
thank you.
Thank
Okay,
you.
Dr.
Thank you.
Bina. Thank you. Thank you very much. So
So
now,
now can I quit, sir?
yes, yes,
Hmm.
yes. Piyush, want
Sir,
to
yes, sir. Sir,
announce?
we can have 5 minutes break so that participants can join. Then we'll again start. So
Okay,
all the
okay.
participants are requested to join within 5 minutes. So as we have to start the oral presentations.
Okay, then after 5 minutes, we will again meet.
Yeah. Thank you, sir. Yeah.
Okay.
Yeah. Good afternoon, everyone. As everybody joined, Manoranjan sir, are you there?
Yes, yes, yes,
Sir,
Piyush.
sir, I think you can start now, sir.
Okay, okay. You can start. Piyush, am I start?
Yes, sir. Yes, sir. You can
Okay.
call the name of participants, sir. Yes.
Okay, okay, okay. So, good afternoon, dear participants. I welcome you all in this virtual platform of 2 days national seminar for presenting your research findings and share with us. So, in this session, we have a total of 15 oral presentation, and I call one by one for their presentation. So, in first OP, oral presentation will be delivered by Aratrik Pal, and his title of topic is on the discovery of two new species of family Lindernaceae. Aratrik Pal, you may start.
Thank you, sir. A very good afternoon to the chairperson, all of my teachers, and the participants. Today I am going to present a seminar on the discovery of two new species of Lindernaceae. Now, the Lindernaceae is a comparatively recently established family based on phylogenetic study. The several genera like Bona, Lindenia, Vandalia, Torrenia, which were earlier treated as members of Scrofulariaceae, now are collectively called Lindernaceae. There are over 250 species worldwide under 21 genera. Now, coming to the new species, uh, one is Bona gracilis, which is described in 2021, and it is a more widespread species throughout South Asia. Another one is Torrenia siliguriensis, which is described from eastern Himalaya, and it is restricted to Siliguri subdivision of Darjeeling district. Now, the coming to the genera Bonaya and Torrenia, we can see in the screen that Bonaya having the calyx segments deeply divided, whereas the Torrenia, the calyx segment is shallowly low. Another one is the number of stamens in Bonaya is only two, whereas in case of Torrenia, there are two pair of stamens, and a pair of stamenos is also present in Bonaya. Uh, and next is the length of the capsule in case of Bonaya is far exceeding the calyx, whereas the capsule is much shorter in case of Torrenia. Now, discovery of the Bonaya gracilis. In 2000, in November 2020, I found a unknown Bonaya species from Siliguri subdivision of Darjeeling district. Now, when I came across the came across the specimens of Torrenia gracilis in Kew Virtual Herbarium, I found it is the perfect match for our specimen. And the Torrenia gracilis was published by Bentham in the Wallis numerical list without any proper description. And later, Bentham synonymized it with Bona verminifolia, which is a synonym of Bona antipoda. Now, the herbarium seeds of both the species, that is Torrenia gracilis and Bona verminifolia, were different based on the position of the pedicels, the size and shape of the bracts, and the shape of the leaves. Now, when we see the which illustration of Bona verminifolia, we found the illustration is exactly illustrating the same species we found from Siliguri, and uh, clearly a species not of Torrenia but Bona, and it also differs from the illustration of Hillcock's uh, Bona antipoda uh, by having dissimilar size and shape of bracts, whereas the size and shape of the Torrenia gracilis is, or the illustration are similar. Now, so based on the its characters, we shift its treatment to Bona, and then we publish it. Now, from our fresh specimens, the difference between Bona gracilis and Bona antipoda is clearly visible. In case of Bona gracilis, we can see the bracts are very similar, and whereas in case Bona antipoda, the bracts are dissimilar in size and shape also, and also the pedicels sometimes are opposites to leaves and sometimes axillary, whereas always in case of Bona gracilis, the pedicels are opposite and with subtended by bracts. Here is a table where we provide the difference between Bona gracilis, Bona antipoda, and Bona sanpabulensis, which is also a recently discovered species from Southeast Asia in 2014. Here is the images of the Bona gracilis uh, taken from our paper. Here is another image showing that difference between Bona dissected flower of Bona gracilis and Bona antipoda. We can see the upper lobe of Bona gracilis is oblong, whereas in case of Bona antipoda, the lobe is ovate, and other lobes of the lower lip are also of dissimilar shape. And the staminodes in case of Bona gracilis are very densely hairy, and in case of Bona antipoda, they are subglabrous. So we published it, and it was published in 2021 as transfer of Torrenia gracilis went to Bona as Bona gracilis espinova. Now coming to discovery of the Torrenia siliguriensis. In October 2020, we found another Torrenia species, unknown Torrenia species. From the image, we can see the species is very similar to Torrenia crustacea, but when it is in field, we can see the flowers of this Torrenia species is much smaller, and where it is to see, and the plants is also much smaller than the Torrenia crustacea. When we dissected the flower of this unknown species, we found that there are many more characters differing Torrenia crustacea, like the lower lobes of the flower is obtuse in case of that unknown species, and in case of Torrenia crustacea, they are subtruncate. Also, the anther shapes are different, and the some glandular hairs are present in case of Torrenia crustacea, which are absent in case of Torrenia, that unknown Torrenia. There are also characters of fruits which differ. We can see from the image that Torrenia crustacea fruit is somewhat ovate, whereas in case of Torrenia siliguriensis, this is more oblong, and the disc is protruting upwards in case of that unknown Torrenia. So we come to a decision that it is a different species, and we bring some more difference between the species shown in this table from our published paper, and then we named it from its uh, type locality, that is Siliguri, as Torania siliguriensis, and we published in Annals Botanici Fenesi in 2021. Uh, Torania siliguriensis, a new species from India. Thank you, everyone.
Okay, thank you, Arashid Pal, and congratulate you for this discovery of two new species from your area. And best of luck for your future research.
Thank you,
Now,
sir.
the participants are requested if you have any queries, you may ask to Arashid. Okay, if there is no question, then thank you, Arashid.
Thank you,
Uh,
sir.
Now I would like to invite our next speaker, Dr. Mudumita Mojumdar, and her title of talk is Medicinal Orchid Conservation and its Sustainable Utilization through Tissue Culture Approaches. Dr. Mojumdar, please start
Yes,
here.
yes, sir. Good afternoon, everybody. Myself, Dr. Mudhmita Mujumdar, Assistant Professor, Department of Botany, Raidighi College, Raidighi. I am thankful to the organizing team for giving me this opportunity. And my topic for today's oral presentation is medicinal orchid conservation and its sustainable utilization through tissue culture approaches. The orchid family is one of the largest flowering plant kingdom with about 25,000 species. Orchids are an ancient group with their present distribution suggesting that their origin probably predates the breakup of Gondwana land about 125 million years ago. Orchids are absolutely ubiquitous in nature and occurs on all vegetated continents. The major orchid-rich phytogeographical regions of India are peninsular India, northeastern India, and eastern and western Himalayas. The father of medicine, Acharya Charak, has brought Indian orchid into world notice way back in 100 AD. India is one of the 12 megadiversity country in the world. According to the Indian Red Data book published by Botanical Survey of India, 10% of the total flowering plants of India are endangered. Next, I will discuss about the ornamental aspects of orchid. Orchid flower is not only beautiful, but also it is long lasting. That is, we can keep it as it is for 1 month in flower vase. They are also very costly and it has a big global demand at market. These are some orchids with beautiful and costly flowers. Owing to their undeniable floral splendor, it comprises a major floricultural crop. Several types of essence are also available from orchid. The common essence, like vanilla, is used as an additive in cakes and ice cream. Next, I will focus about the medicinal importance of orchid. Orchids are generally used in indigenous system of medicine. They are the source of important secondary metabolites like dendrobium, nobiline, and erionin. so many, and it has some therapeutic potential that act as an antioxidant, anti-angiogenic, and anti-tumor agent. Asthavarga is a group of 8 drugs in Ayurvedic system which are used for the presentation of tonic such as Chavanprash, which consists of 4 orchid species like Malaxis muscifera, Malaxis acuminata, Habenaria intermedia, and Habenaria edgeorti. The use of orchids in herbal medicine has a very long history. There is not a single ailment for which therapeutic and curative elements are not found in one or the other orchid species. It is also reported that orchids were held great repute as a medicine with strengthening and demulcent qualities in Ayurveda. Now, I am going to talk about the therapeutic utility of some medicinally important orchid. This is a picture of Aerides odorata, where the tubers of this orchid has the eridin, which is one of the important phytochemical used in the treatment of skin infection and fever in children. This is a Nedrobium nobili. Here, the whole plant is used for the pulmonary tuber treatment of pulmonary tuberculosis, general debility, and anorexia. This is a picture of rhinchostylis. I just, sorry. I think this is, use just a minute. This is another medicinal important orchid, that is Vanda tessellata, that is Dactylohateria. This is a Malaxis muscifera, which is used in bleeding, burning, sensation, and the fever. And Rhinchostylis retusa, whose leaves of these plants are used in cure the rheumatic disease. We all now I coming to the point, that is the orchid rarity. There are numerous varieties of orchid. Some of them are on the path of extinction due to the various natural factors as well as anthropogenic factor. The most important natural factors are confinement of orchids to a limited area, several biological factors, and the natural calamities like forest fire, flooding, etc. The two important anthropogenic factors are the overcollection as well as habitat destruction. They are mainly responsible for the orchid rarity. Next, I will say about the orchid conservation of orchid. I have discussed the importance of orchid and rarity also. At the same time, due to the uh, great commercial potentiality, I it's a we can earn money, provide the employment. Therefore, the orchid should be conserved essentially for multivarious purposes. Conservation can be done by the two approaches, that is the conventional approach, that is in situ and ex situ conservation, and in case of biotechnological approach, in vitro conservation technique is mainly adopted. I will now speaking about the in vitro conservation. Orchids can be grown and conserved in test tube for prolonged period, that is called the in vitro conservation, just like the test tube baby. Under in vitro condition, there are three basic methodologies generally followed: in vitro propagation, in vitro storage, and cryopreservation. Generally, you know that the plant propagates through the seeds, which is a product of sexual process. But in case of orchids, seeds are not so reliable for large-scale propagation. Although they produce millions of seeds, but they do not germinate naturally. Therefore, an alternative method of in vitro propagation called micropropagation has been adopted for the same purpose. So, what is micropropagation? Micropropagation, it is an asexual vegetative propagation of plants using the in vitro technique. There are so many techniques like organ culture, shoot tip culture, callus culture, direct and indirect somatic embryogenesis. But basic technique for successful micropropagation follows the five important stages like preparative stage, initiation and establishment of aseptic culture, proliferation of shoots and shoot bud, rooting of in vitro formed shoot, acclimatization of plants and field transfer. Orchids can be conserved by several methods of storage, like medium-term conservation under minimal growth condition and medium-term conservation using growth inhibitor. We know that plant can grow from natural seeds which are available from the plants in nature. But where seeds are not viable or recalcitrant in nature, in such case, scientists have produced the artificial seeds which are as good as natural seeds. Such seeds could be stored under the sterile condition for prolonged period, or it can be stored in liquid nitrogen where the temperature is more or less minus 196 degrees centigrade. This process is known as the cryopreservation. And this is the schematic diagram of which is employed for the in vitro storage of the orchid protocol. So in conclusion, we can say that orchids are mainly regenerated for their herbal drugs, and advanced countries are earning in millions by establishment of orchid industry. Their cultivation on a large scale using modern techniques such as tissue culture are absolutely essential to meet their increasing demand. The techniques are used to preserve rare and the threatened orchid germplasm. Very little effort has been made to cultivate the medicinal orchid for the commercial purpose. So, achieve the ultimate goal of the sustainable use of the medicinal orchid, extensive research is still necessary. And plant tissue culture could be one of the most suitable alternative tools to minimize the pressure on natural population of medicinal orchid and their sustainable utilization. Thank you.
Okay, thank you, Dr. Madhumita Majumdar, for your for sharing your research outcomes on the medicinal orchids and their conservation, along with the sustainable utilization through tissue culture approaches. So, anybody have any question to Dr. Madhumita Majumdar? Then you may ask her. If there is no question, then we have to proceed. And now I would like to request Dr. Devabroto Das, and his title of topic is Project Medicinal Plants of Jitka Forest and Conservation Practice in Jhargram district of West Bengal. Dr. Devabroto Das, are you there?
Yes, sir. Yes, sir.
Okay. Then you please start your presentation.
Yes, sir.
Okay.
Good evening, everyone. My topic is medicinal plants of Jitka forest and conservation practice in Jhargram district of West Bengal. You know, the Jitka forest is famous because during Maoist battle in the year uh, 2008, that is between Marx left front, Marxist versus police, and that is Maoist. So this is famous. The jungle is famous as that is Jitka forest, and this is before the forest of the Lalgod. And from the Midrapur to Lalgod, one forest is Bhadutola forest. So a large patch of fallow lands present here and intermingles with varieties of jungles up to the forest of Jharkhand. So it is the lower tract of the Chotnagpur plateau, and the forest is dry deciduous sal forest. So my research point is on the forest of the lateritic sal dominated forest. A large number of forest resources are available, in various parts of our country. Patterns of collections and use of forest resources are different. Even the processing and preservation techniques are also different from one side to another side. All natural medicinal products are used directly or indirectly for the use of local people in various purposes. Jitka forest is of Southwest Bengal forest under the beat of Jitka and a range Lalgod under the division of Jhargram. Such quality natural products are available which are in the forest union, that is Jitka up to Lalgod. In this communication, I discuss 20 very important medicinal plants and the direct use which have been presented under the jurisdiction of Southwest Bengal. Here actually the significant role played by Joint Forest Management Committees, that is JFMC, and their importance remains and they are involved to make the system sustainable. Even they serve as a master ruler to run the process smoothly, even to manage the biodiversity. Here, Jitka is a forest, Ralgod is a forest, Rangamati is a forest, Purnapani is a forest, Chandra is a forest, and other two villages are Baghura and Jitka village. One interesting point is that Jitka forest having tribal community, particularly Lodhas. Lodhas are the important tribal groups in our country. Other heart, this means that is weekly markets of Lalgod and Bhimpur has been taken into consideration for our study. In this map, I am showing that this point is that is the Lalgod part under the jurisdictions of Midnapur. The Midnapur districts are divided into two districts, that is Jhargram and Midnapur in the year 2017. So important point is that particularly on Jhargram forest and in Jitka forest. Regular and periodical surveys for consecutive 3 years, 2016 to 2019, have been conducted in 4 areas, namely lateritic forest, villages, local markets, and weekly markets, that is called HART, in and around the forest to collect usable parts of medicinal plants and nature, that is natural products of the forest kind. Medicinal plants along with the non-timber forest producing plant samples are collected from the selected marked small plots, that is within the forest. 1 into 1 meter square for herbs, 5 into 5 meter square for shrubs, and 10 into 10 meter square for trees at regular intervals. The study was conducted for consecutive 3 years. Field trips were arranged to cover all the study sites in random sampling method in Jitka forest of Southwest Bengal. I have taken other that is related forest sites to compare the productivity and that is huge value of all the potential non-timber forest producers as medicinal importance. The ethnomedicinal and medicinal values of plants are collected through cross interviews on local people. The matched that is parts with literature published time to time, and local and common names including usable plant parts and photographs are taken to verify the authenticity of species and make record data. And identification of species was done with the help of available literature, herbarium specimens kept in museums of various institutions, even my college herbarium section. Present study reports 20 very important plant species and their direct value from Southwest Bengal, which have been recorded from the Jitka Forest jurisdiction of West Bengal. I am coming to the table 1. The significant role of the Joint Forest Management Committees are immense. and they are involved to make the system sustainable. Even they serve as a master ruler to run the process smoothly, even to manage the biodiversity in the said area. In the table, various types of plant-based items have been presented, like whole plants, roots, barks, flowers, fruits, and seeds used for medicinal purpose. Here, these are the medicinal plants, amla. This is not forest species in our Jitka, but aside the forest, that is in degraded lands, amla are available, and this is a wild kind. Mohul is important, as probiotic. Mohul flowers, dried one is important, and in the market, the value per kg is 60 to 70 rupees. And Horitoki, that is Terminalia chibula and Boheda, all are very important medicinal plants, and they used and they sell in the market. And from the nodal market, it is going to the other markets. Duthilata or Anantamul, scientifically Hemidesmus, Isormul, Aristolochia, Sothmul, Asparagus, Comarica, that is Smilex, Macrophylla. The important plant parts of these plants are fruits, roots, and in case of seeds, the bijasal, Pterocarpus marsupium. The species is available in the forest, scanty areas, and the availability and frequency is very, very low. So to know the actual occurrence of the mother plant species, to record the plant to get the seeds for further degenerations. And as the plant having no regenerations below the ground level, so that is important one. And Koronja, this is not entirely into the forest patch, but aside the forest, there is spinata are common. Whole plants like kalmeg, enormous volume of kalmegs are marketed from the forest, and flowers like lothpolas, polas, athmojada, and simul, also dhadika or abata. Barks like orjun, jamun, neem, aswan, etc. are available in the forest. So my discussion is that Jitka forest is a sal-dominated forest under the jurisdictions of Lalgod forest, a range of Jhargram forest division in West Bengal. Sal forest is a deciduous forest in the dry lateritic tract of lower plateau in Chotanagpur, while the continuity tends up to the neighboring areas in Jhargram district. Soil pH of this forest varies from 6.2 to 6.8, and the temperature ranging between 7 to 29 degrees centigrade. The huge luxuriant growth of green vegetation found in the forest during monsoon to late monsoon. A large number of herbaceous plant species have been recorded here. Not only the herbaceous species, you can find shrubby species and tree species which occupy a good role in the fulfill of quality yield in terms of medicinal products. Tribal and marginalized people, I say earlier that Lothas and Santals, but Lotha community are very, very, that is distinct in the Chitka forest. If you go to the Sarpanchayat, that is election, then the record revealed that in 2019, only 19 family members are there in the Chitka village. These plants, that is tribal community managed for their daily purpose as non-timber forest produce, though a large number of medicinal plants they use for commercial purpose. Forest management committees and local people's participation play a crucial role to make the forest productive and managed in a sustainable way. Lotha is the main tribal group of Jitka forest. Of tribal group of the peoples are found here with other groups. They protect the forest for their own purpose, though they maintain a scientific approach directed by forest and peoples of scientific community. Here, main problem is the forest fire. Here you can see the figure 1 to 4, that is the Ramnathon, that is also Milex. That next one is Lothpolas or Butia superba, then Terminalia, that Terminalia species, that is Aswan, then this one is Horitoki, Terminalia chibula, and this is Terminalia belerica, and then Dhadika, and lower left one is Helicteris isora, then this one is bitter king, that is Andrographis paniculata, and last one is Aristolochia indica. So my conclusion is that result revealed from this study, there are high demands of the medicinal plant products from the forest. High diversity value for natural products as non-timber forest produces are available there. These are whole plants, root, barks, flower, fruit, seeds, and resins. Resins, that means sal dhuna, in Bengali form sal dhuna. These natural products are used as a potent source of economy as well as a good source of chemical constituents to develop various types of industries in and abroad. Main use is here to develop local medicinal plants garden by local people for income generation, not only depending upon the forest but of their own garden and improved by other, that is institutions or NGOs or charitable societies or forest department or other department. Our duty is to promote them and encourage them to do it for better management, for the better that is society. According to Forest Department report, Jitka people use 2 days, that is February 16th, 25th February and 16th March, sorry, 25th February and 16th March as hunt days and use Jitka forest as their festival ground. Now they use the forest not only for hunting purpose but collect many valuable plants, that is medicinal plants, for their use around the year in a managed way. And my acknowledgement, that is the organizers, Advancement of Plant Science for Food Diversity and Nutritional Security, Department of Botany, North Bengal University, in collaboration with UGC, RDC Cell, Forester, Southwest Bengal, Director BSI Kolkata, Principal Chief Conservator of the Forest, Government of West Bengal, DPI, Harvesting Department, Government of West Bengal, and my officer in charge, Lalgod Government College. These are the references. Thank you.
Okay, thank you, Dr. Devabroto Das,
Thank you, sir. Thank you.
for your elaborative talk on medicinal plants that are found in Jitka forest. So, is this forest is natural forest or plantation forest?
No, sir, this forest is natural forest, salt dominated natural forest.
Natural forest. Okay, so in your materials method part, I have seen that you have performed quadrate for 1 by 1, 5 by 5, 10 by 10. For what purpose you did this?
Sir, for productivity study, not all tables and not all data are here. Actually, to quantify the actually production, that is assessments of biomass and assessments of important medicinal plant parts and important products for quantification study.
So, any measures they have taken towards the conservation?
Yes, sir. Yes, sir. At first, I told you that Bijasal or Piasal is important one. 4 or 5 trees are available in the forest, but no regenerations just below the ground level, which I think need very immediate. I think very immediate attention may be made to that is reproduce all plants after the preparations of seeds. Like other species, many more are there.
Okay, thank you. Anybody have any question? Okay, if not, then thank you again, Dr. Das, for your nice presentation. Now, I would like to request Dr. Kishore Vishwas to present his topic on phytosociological analysis of natural vegetations in Duars region of West Bengal, India.
Okay, very good afternoon to all. With the kind permission of the chairperson, I would like to present my topic. But before that, I would like to share, please allow me to share my screen. Okay. My topic is phytosociological. Am I audible?
Yes, yes.
Hello.
Okay. You are audible.
So,
you can proceed.
My topic is phytosociological analysis of natural vegetations in Duars region of West Bengal, India. Generally, the entire resource-rich plain of West Bengal and Assam around Bhutan is known as Duars. And this is the sub-Himalayan foothill region and extends from Nepal to Assam and is bordered by hills of Darjeeling and Bhutan to the north and Coach Bihar, North Dinajpur and Bangladesh to the south. And this Duars is one of the most resource-rich resource-rich center in the Bengal. And this belt is contiguous with the Darjeeling Himalaya, which is in turn a part of Himalaya hotspot of biodiversity conservation, and that is recognized by IUCN. And being contiguous with the Himalayan hotspot of biodiversity conservation, these terrestrial belts, especially the Duars, is rich in phytodiversity and their diverse habitat. And this supports a huge number of rare, endemic, and threatened elements. That's why a number of protected areas like national park, wildlife sanctuaries, reserve forest, etc., has been established in this belt. But the vegetation of this, especially the natural vegetation of this belt, is now under different type of threats. So my present objective of my study was to characterize the natural vegetation, to understand the phytodiversity in natural vegetation, to study the seasonal variation in species diversity, composition, and community structure, and to study the phytosociological aspect of the natural vegetation. So this is the methodology. I generally adopted the three-tier nested quadrant sampling method, and this is the layout of three-tier nested quadrant. And these are the formulae which I have been used to analyze the phytosociological data. Generally, frequency, relative frequency, density, abundance, IVI value, and different biological indices have been calculated. So, for the present study in Duars, mainly the Lataguri and North Rajabhatwa region has been selected for sampling, and all the tree, shrub, and herb layers in three consecutive seasons, that means winter, post-monsoon, and pre-monsoon season, was selected for the study. And this table shows the number of recorded species, genera, and families recorded from different layer of different sites. That means tree layer, herb layer, and shrub layer in different seasons. And this is the graphical presentation of the species, genus, and family recorded from different from these two sites. And generally, the post-monsoon season was found to be recorded for the highest number of species, genus, and family, and that was same for all the NRBK and Lataguri site. So tree layer, this table actually shows top 10 species of tree layer in winter season, having their IVI values, and Tabani-Montana diabeticata was recorded for the highest IVI values, that was 17.983, and was followed by Dendrocnidis sinuata, Mallotus nudiflorus, etc. And depending on that IVI values, dominance diversity curve was prepared, and this curve shows that dominance diversity of this of the tree layer in NRBK site in two seasons, that means winter and post-monsoon season. And generally that tree layer shows very little difference, or actually we can say there are no differences in this two layer. Only the differences was found in the number of individuals only, and that may be due to the changes in girth, DBH, or diameter in girth height, diameter in breast height. And this is also same for the shrub layer, and Coffea bengalensis, that was recorded for the highest IVI values, and was followed by Clerodendron infortunatum, then Tabani-Montana diabeticata, and all. And here is also the dominance diversity curve of tree layer, that means winter, pre-monsoon, and post-monsoon. And here a little difference was found in the diversity curve, dominance diversity curve of this tree layer, that means 3 seasons, sorry. Then the herb layer, it was dominated by Chlorantha ciliator, and was followed by Mycenia micrantha, then Archaeospermum wallichianum, Phallopsis imbricata, and all. And this is the dominance diversity curve of herb layer in 3 seasons. Here, the post-monsoon season differ from winter and pre-monsoon season because of that luxurious growth of ground cover vegetation in due to the favorable growth condition in post-monsoon. So this is the Lataguri record for Lataguri site. Here the Lataguri that means the sampling site showed the dominance value of highest dominance value of Bacillus glaucosens. Then was followed by Bauhinia variegata, Stereospermum tetragonum, etc. As it was tree layer, similarly in like in other earlier cases, the tree layer shows tree layer in winter and Post monsoon shows very little difference. This is the shrub layer. It was dominated by Mycrania micrantha. Generally, in my study, the climbers were considered in shrub layer, and it was followed by Justicia datuda, Clerodendron fortunatum, and this is the dominance diversity curve. Here, the dominance diversity curve also shows differences in tree season, and the shrub layer in Lataguri site actually showed little heterogeneous condition of the vegetation. That's why it shows somehow steeper type of curve. Then half layer, it was dominated by Mycini micrantha and was followed by Opisthomas barmani, Agiritum hostrinianum, Piper betelodis, etc. And this is the dominance diversity curve of the half layer. And these are this three the graph for the different diversity indices, mainly the species diversity index, concentration of dominance, and species richness of tree layer, shrub layer, and half layer, and in different season, in winter, post monsoon, and pre-monsoon. And in case of tree, they generally two season. Actually, the little difference was found in three season, in post monsoon, pre-monsoon, and winter. That's why only the two season has been represented. And tree species, regarding the species diversity, that tree layer was found to record a lesser value of species diversity index, and much lesser concentration of dominance, and higher concentration, higher value of species richness. That means it indicated the tree layer was tree layer or canopy layer was having the lowest diversity and lowest concentration of dominance, and higher value of species richness. So major finding of my study is that the entire dwarfs, natural vegetation of entire dwarfs was very rich and diverse in phytodiversity, and all the trees, shrub, and herb layers supported a huge number of species. Regarding the diversity, the herb layer was in the topmost position, and in the tree layer was the lowest position. And tree layer was, as I mentioned earlier, that it has a very high richness and lowest value of concentration of dominance, and differences in number of individuals only found in the tree layer in different seasons, and that may be due to that changes in girth height, that means diameter in girth height. That is the main criteria for considering tree, herb, and shrub. And variation in species composition also found in tree layer, and that was recorded mainly due to the anthropogenic activity like cutting and felling. The shrub layer, it also was recorded for higher diversity and richness value, and lower concentration of dominance. And herb layer was found to be very diverse and having lower value of richness and highest concentration of dominance. That means somehow heterogeneous type of condition, and that condition was prominent in post monsoon vegetation because of luxurious growth of one or few species, one or few herbaceous species. And some traits were also recorded or during the study, like invasion by exotic species, then initiation of plantation, and some other type of anthropogenic activities. So these are the selected reference. Thank you.
Okay, thank you, Dr. Biswas. I have one question. Why
Yes, sir.
you have chosen that winter and post monsoon? I think both the time is same. It will start from November, December to January, like that.
Yes, yes, sir.
So,
Actually, sir, I have in case of tree layer, I have selected only true. That means winter and pre-monsoon, winter and post-monsoon, and that was mainly due to trace any kind of anthropogenic activity like cutting, felling, and some other type of activities.
but you have discussed about the areas those are inside the forest, national park,
Yes, yes,
where
sir.
tree cutting, falling is not possible.
Yes, sir. It is possible, sir. I
No, no,
have
no. This is due to accidental means or by that is calamities. This is the other thing. But my question is, you have chosen winter and post monsoon. So
Yes,
winter
sir.
and post monsoon is the same period. That is November, December, January. Why you left the monsoon period and pre-monsoon? That is the April or the month of July. Because in monsoon there will be the maximum number of species are there. If we leave that tree part, then shrub and herb layer, you will get much more species. And if
Ac-
you see your that is calculation, there you will find that insignificant result, that is the 6.9 in winter, and post monsoon it is 7. So point 0.1. It is not significant. Better you have to choose during the pre monsoon or post monsoon, or winter or summer. So I think it will give a better result. Anyway, thank you for your presentation. Okay.
Okay, sir. Thank you for
Okay.
your
Okay.
suggestion also.
We
Thank you for your
will
suggestion also.
okay. Thank you. Now our next speaker is Devobroto Modo, and his title of presentation is Dry Neria quercifolia, rhizome, methylonic extract ameliorates rheumatoid edema in Freud's complete adjuvant induced arthritis in rats. So, Devabroto, are you there? There?
Yes, sir.
Okay, so you may start. You may start.
Thank you, sir. Good afternoon to respected chairperson, sir, respected teachers, and other fellow. Today I am here to present my seminar topic on Drinaria quascifolia rhizome, methanolic extract ameliorates rheumatoid poedema in French complete adjuvant induced arthritis in rat. And this work has been carried out under the supervision of Professor Shomen Bhattacharya from Department of Zoology, and the co-supervision of Dr. Subarna Thakur from Department of Bioinformatics, University of North Bengal. First, come to the introductory part. As we all know, that is the inflammation, the body's own defense mechanism, is a very common biological cascade. And in response to any kind of foreign pathogens or tissue injury or any kind of chemical irritation, and it acts by removing those injurious stimuli, and it begins the healing process, and which is very rapid and self-limiting. And both the infectious and non-infectious stimuli can trigger the
Hello, Devabroto. You are not audible.
I think, sir, he has left. Yeah, pardon.
Yes, Piyush?
He's there or not? I don't sure.
I think he is facing some issues.
You can tell the next one.
Uh, Hello?
Hello. So
Deva,
I'm saying,
Devabroto, are you there? I think he is trying.
yes, sir. Okay, 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 sir.
hello.
Okay, Devabroto,
Yes, sir.
you present your work.
Okay. Sir, is it audible and is it visible?
Yes, yes.
Yes, thank you. And both the infectious and non-infectious stimuli can trigger such inflammatory pathway, or that is the acute inflammatory response, and which mainly restoring the physiological process. However, when this acute inflammatory response become prolonged, and it may chronic, and it enhance extensive tissue inflammation, and contributes to a variety of chronic inflammatory diseases like type 2 diabetes, asthma, ulcerative colitis, and even rheumatoid arthritis. People with this condition often have higher level of inflammatory markers in their bodies that helps to maintain those prolonged inflammatory state. So it is very essential to identify and manage those particular inflammatory consequences to prevent the further complication, and to help the restoring in the physiological homeostasis. And to treat these consequences, commercially available anti-inflammatory drugs like non-steroidal anti-inflammatory drugs, opioid, even steroid often used. But these drugs act like only alleviate symptoms, treating or resolving particular symptom. And additionally, long-term uses is associated also with several side effects like drug dependency, dizziness, gastrointestinal problem, impairment of hepatic and renal function, cardiac disease, and even sometimes it can hamper the normal neuronal regulation. Um, in this scenario, natural products are emerging as the therapeutics because of their obviously the pleiotropic nature, their availability, their economicability, and having side effects. Um, Now, plant that is the Drinaria quercifolia or Aglamorpha is a very well-known territory, and it is known as fern, and it is also commonly called this functional grass in Bengali, and it is also medicinal plant which have various diseases. The front are used to use different varieties of influenza, foliage diseases. The from the rhizome is among the hills of eastern Tamil Nadu get from rheumatoid campaigns. Even the tribe sub-Himalayan is like Toto also there are region they used quercifolia to promote the treatment the bone fracture to strengthen the bones. So initially non-evidence evidence medicinal use of rhizome extract.
Hello, Devabrata, are you there? Okay, I think Devabrata is facing some network problems, so he must be present later on. Now we have to proceed, and now I would like to invite next speaker, Ravi Prasad. Are you there?
Yes, sir, I am
Okay,
here, sir.
Ravi, welcome. Okay, you may present your talk.
Okay, sir.
Uh,
Just a minute. I am sharing my screen.
his topic of presentation is important medicinal plants of Sariksa Tiger Reserve, Rajasthan, India. So Ravi, please.
Yes, sir. Yes, sir. Am I audible and my slides are whether visible?
Yes, yes, yes. It is
Yeah.
audible. You are audible and visible.
Yeah. Good afternoon to all of you. Myself Ravi Prasad, and today I am going to talk about the important topic, that is the important medicinal plant of Sariska Tiger Reserve, Rajasthan. Our general perception about the state of
Ravi, you are not audible.
Hello,
Yes, yes, yes.
sir, whether my voice is coming or not?
Now coming. Earlier it was not
Okay,
coming.
just a second, sir. Just a second, sir. Yeah.
Yeah.
Good afternoon to all of you. Myself Ravi Prasad, and I'm going to talk on an important topic, that is the important medicinal plant of Sariska Tiger Reserve, Rajasthan. Our general perception about the state of Rajasthan is that in Rajasthan we will get only thorny and shrubby species, but the actual scenario is not like that. So today I'm going to take you to the medicinal plant journey of the Sariska Tiger Reserve. And this is my study area. Sariska Tiger Reserve is situated in the Alwar district of Rajasthan, in the northeastern part of Rajasthan, and the total geographical area of the Sariska is 1213 square kilometer. It is a big tiger reserve. The vegetation of the Sariska corresponds to the northern tropical dry deciduous forest and northern tropical thorn forest. As you can see from in this slide, the Sariska is having a very good vegetation. It is having a some desert element, some sand dunes. At the same time, it is also having a wetland like situation, scrub forest, and at the same time, it harbors a number of medicinal plants. And in this slide, these are some of the important medicinal plants of the Sariska Tiger Reserve. I am working on Sariska since last 4 years as part of my PhD work, and during my exploration, I have collected number of medicinal plants from that area. And in this slide, you can see the first one is the Abrus precatorius. The seeds are quite poisonous, but after several rounds of boiling, it is used for different medicinal purposes.
Ravi, your sound is not coming. You are not audible. Are you there?
Hello, sir.
So you wait. Okay, you just wait few minutes. I will call you. Yes, Ravi. He is also facing network problem. Devabrata, your network issue is resolved now.
Yes, sir.
Okay, then you go. Then you start. Okay.
Okay, sir.
Ravi Prasad will finish after you.
Okay, sir. Sir, is it visible?
Not yet.
Okay, sir.
Devabrata, present your work, your slides.
Visible?
No, no.
Hello, sir. Sir, the student is not screen. Can present the screen, my screen.
Wait to come.
Sir, I can't find my. Is it
Okay, you present later on. Huh? You stop sharing.
okay? Okay, sir.
Now I would like to request Pavitra Patil. Are you there, Pavitra Patil?
Yes, sir.
Yeah. So, and her title of presentation is a synergetic combination of leaf extracts and solvents as the natural health, enthalpy control agent and experimental study. So, you may start, Pavitra Patil.
Okay, sir. So is it visible?
Yes, yes.
Okay. Good evening, everyone. I'm Pavitra, and I'm presenting my topic on a synergetic combination of leaf extracts and solvents as the natural anthelmintic control agents, an experimental study. A pesticide is any compound or formulation used to control pests which contain active ingredient and other substance to aid its delivery to target organisms and hence minimize the pest. For example, herbicides, insecticides, rodenticides, etc.
People believe that natural pesticides are always safe and more eco friendly than man made or synthetic pesticides. Helminthic infestants are now being recognized as a cause of chronic ill health and sluggishness among the children. Worms have the ability to destroy young and tender plants. Also, when they are present in large number, they destroy the grasslands by making too many burrows in the soil. One practical way of developing cheaper and effective anthelmintics is to study the indigenous herbal remedies. Material and methods. The plant materials used in the experiments are Iconia crassipus, Carica papaya, and Moringa oleifera. The Iconia crassipus is commonly known as common water hyacinth, and it belongs to the family Pontaderaceae, and it's an aquatic plant collected from the Unkal Lake, Hubli, Karnataka. Carica papaya is commonly known as papaya and belongs to the family Caricaceae. It's a medicinal tree collected from Basveshwanagar, Hubli. Moringa oleifera is commonly known as drumsticks, belongs to the family Moringaceae. It's a traditional medicinal plant collected from PC Jaban Science College, Hubli. These were the location of map showing the study area. Basveshwanagar, Hubli, Unkal Lake, Hubli, and PC Jaban College, Hubli. Plant extraction. We have collected the plant from the different places of Hubli, and the plant leaves were washed with the tap water followed by the distilled water. After the surface sterilization, we kept the plant leaves in a shady area for around 15 days for the drying. After the drying, we have powdered the plant material or the plant leaves by locally available grinder. After the preparation of plant powder, 5 gram of each plant powder is added to the 50 ml of ethanol, acetone, and petroleum ether separately and kept it for digestion for 24 hours with the occasional shaking. After the digestion period, it is filtered with the help of Whatman filter paper. These were the images showing collection of plant material, drying of plant material in a shady area, weighing of plant material, and the prepared plant extract. Anthelmintic assay. The prepared plant extract is now used for the anthelmintic assay. The plant extract were treated to the Petri plates containing the Whatman filter paper. Here, albendazole is granted as the positive control and the solvents are granted as the negative control. The earthworms were introduced into the pre-treated Petri plates and observed. The paralyzed and the death timings were noted. The death of the earthworm is confirmed by the change in its size and its ceased movement. This is the vermicompost pit from which I have collected my Perithema postuma or earthworms, and this is the microscopic view of the Perithema postuma. Results. The experiment have done by using Iconia crassipus, Moringa oleifera, Carica papaya, ethanol, acetone, and petroleum extracts against the Perithema postuma. This table shows the paralysis and death rate and as well as the mortality rate of the anthelmintic activity. Here, the highest rate I have gotten Iconia crassipus ethanol extract, that is, the earthworm get paralyzed at 25 seconds and it died at the 28 seconds here and the lowest rate was found in Carica papaya petroleum ether extract, in which the earthworm get paralyzed in 35 minutes and the death occurred at 52 minutes. Here almost all the plants has given the 100% mortality except the Iconia crassipus petroleum ether, in which the result were negative. And this is the graphical representation of Perithema postuma. Graph 1a shows the paralysis rate and graph 1b shows the death rate. Here we can see the petroleum ether extract has taken the most lengthiest time to give the results compared to the ethanol and acetone extracts. These are the images showing Petri plates containing Iconia crassipus ethanol extract with Peritima postuma, and figure 8 shows the Petri plate containing Carica papaya acetone extract with Peritima postuma. Conclusion: people need to end the habit of using harmful pesticide, and they have to switch to the biopesticide, which can break down quickly in the sunlight and in the soils. Biopesticide can easily be washed from the fruits and vegetables, making them healthier and safe. Based on high mortality rate and highest death period, it is concluded that the application of Iconia crassipus, Moringa oleifera ethanol, and acetone extract as a plant-derived vermicides on Peritima postuma can control the damage. Hence, concluded that the ethanol and acetone plant extract are more suitable for the preparation of biopesticide as compared to petroleum ether plant extract. So, ethanol and acetone solvents are more potent than petroleum ether solvent. These are my references. Thank you.
Okay, thank you, Pratibha, for your presentation. Anybody have
Thank
any
you, sir.
question? Any queries?
I am a Pompey Ghosh. I have one question.
Please.
That is,
Yeah, yeah.
Feituma postuma is not a paste at all, as per my knowledge.
Ma'am, it's a
Ma'am, it's
Indian
an Indian
earthworm. It
earthworm.
it will
But
cause
it is not
the
paste at all. It's a
sir,
friend of
ma'am.
farmer.
If it is
If it
present
is
in
present in
large
a
number,
large
it
number,
will
it will
cause so
cause
many
so many
damages, ma'am.
damages, ma'am. I think not so.
I have
I have
referred so
referred so
many
many
journals, ma'am.
journals, ma'am.
According
But
to that,
according to
it's a
that, it's
pest.
a pest. But farmers and as I as I am doing so many agricultural crops crops in my laboratory, and
Yeah,
I
ma'am.
several time use this as a good thing. Okay,
Ma'am,
Fedma
when they
postuma.
are
When they are
present in
present in
very
very
minimum
minimum
amount, it
amount,
is
it is
helpful, ma'am. If they are present in
helpful,
large number, they can
ma'am.
create
If they are
many
present in
destroys,
large
ma'am.
number, they can create many destroys, ma'am.
Okay, Pratibha, there are lots of pests that need to be controlled. Why you choose these?
Because the earthworms are they have the similar characters. If we know how to control the earthworm, then we will know the similar character of like tapeworm. We can know the strategies to control the tapeworms and other pests. So I decided to do it on earthworm.
But on the other hand, earthworms are called as the friend of farmer.
Yes, sir.
Hmm? Okay, anyway, thank you. Thank you for your
Thank you, sir.
presentation. Uh, Next presenter is Joyati Saha. Are you there?
Yes, sir. I'm here.
And her title of topic is Study on Heavy Metals Uptake and Plant Growth Promotion Potential of Multiple Heavy Metal Resistant Pseudomonas species isolated from arable land of Uttar Dinajpur. So, Jayati
Okay,
Shah, please start.
join, sir.
Janti.
Okay. Okay, sir. Okay, thank you. First of all, Chairperson sir, respected Chairperson sir, and other teacher, my respected teachers, and all of you present here in this virtual platform. And I'm just wait. I'm sharing my slides. Just a minute. Sir, is it visible?
Yes, go for full slide
Okay, okay, sir. Thank you.
presentation.
Okay, sir. Thank you. This is my topic, study on heavy metal uptake and plant promotion potential of multiple heavy metal resistant pseudomonas isolated from edible land of Uttar Dinajpur. With the inception of rapid industrial revolution, several anthropogenic activities like mal-agricultural practices sewage irrigation, wastewater irrigation, and rampant application of chemical pesticides and fertilizers. This has actually led to the substantial release of several toxic heavy metals consistently to the major environmental emitters like soils and waters. And in this regard, actually, as the heavy metals are non-biodegradable and persistent, they gradually accumulate and transfer. In each of the trophic level and hampering the ecosystem. And in case of arable lands and arable soil, this is of utmost important because, in order to meet the increasing food demand, these arable lands are of lot of importance. And the soil contamination in such arable lands not only provide the potential human health risks, but also it decreases the microbial viability and diversity, and hence deteriorating soil health. To overcome this situation, several microbial remediation have been proved to be fruitful in as a cost-effective, innovative technology. And several metal and among the soil dwelling microbes, bacteria deserve special attention in ameliorating the hazardous metal contaminants from the soil. And several soil microbes like Bacillus, Pseudomonas, Acinetobacter, etc. possess, and it is well established that they possess the resistance mechanism, heavy metal resistance mechanism at the elevated level. And due to the metalliferous, due to the continuous exposure or coexistent in metalliferous soil environments, actually they have gradually adapted with such resistance mechanisms like bioabsorption, bioaccumulation, and biofilm formation, etc. Hence, these bacteria assisted remediation in order to control or remove the metal contaminants from the arable soil. This is of this deserves special attention or is of utmost importance. And here this region actually, Uttar Dinajpur and specifically the Raigans block is still unexplored in regarding the diversity of soil microflora in arable lands. This serves an excellent opportunity to consider this region. And not only that, several heavy metal resistant bacteria have been shown to promote the plant growth. Obviously, metal resistance and plant growth promotion ability, these two important aspects actually here has been selected as the potential area to explore. Hence, considering the immense potentiality of heavy metal resistant bacteria in metal removal as well as in plant growth promotion, this study was carried out, focusing on the isolation, screening of bacterial isolates from arable land soil, and then minimum inhibited concentration determination, and screening was done against total 9 different heavy metals, and then MIC was determined, and subsequent growth study, metal uptake analysis through ICP-MS, then plant growth promotion detection, and finally molecular identification. And my study area is arable lands of Uttar Dinajpur, specifically cultivating Tulai Panji rice, the most important indigenous rice variety here. And this area of research is still unexplored. So hence this serves as the excellent opportunity. Next, the methodology that I have followed during the course of my study, sample collection, ICP-AES analysis was done initially to detect the elemental compositions present in the collected soil samples, and then bacterial isolates were obtained, and then biochemical characterization followed by MIC determination, bacterial growth study, and metal uptake analysis, and these are the several potential test, in vitro screening, not potential, in vitro screening test that was followed during the in vitro screening of the isolates regarding plant growth promotion, and finally molecular identification. And this was found that the soil was acidic soil. pH was when pH was measured, it was found that it was acidic, and so these elements, chromium, zinc, copper, nickel, cadmium, these were found in the collected soil sample, and to the standard serial dilution and pore plating and streaking, a total 20 different heavy metal resistant bacterial isolates were obtained, and the screening resulted or revealed that these 20 isolates possess the metal resistance potentiality, not only against single metal but also against multiple heavy metals. These are some culture plates potential regarding the potential isolates, showing the vigorous growth in some elevated metal concentration. First, this is cadmium. These isolates were found to produce bright green pigment, and this is ferrous, as shown in the slide. And next, these are some of the figure of some biochemical test, and some of the isolates here only have been shown. Next, these are total 20 bacterial isolates that I have isolated, and the total biochemical profile. These tests were carried out, and this is the biochemical profile of the total 20 isolates. Then MIC, minimum inhibited concentration, against total 9 different heavy metals were carried out, and each study were carried out or replicated in thrice, and the standard deviation were calculated and shown in the parenthesis here in this chart. And then after MIC determination, this was found that some isolates possess several resistance potentiality against multiple heavy metals, and in some elevated concentrations. Based on the results, total 3 isolates were selected, and this is the growth curve study. Here, growth study was conducted in both normal media as well as these heavy metals. Total regarding growth study, cadmium, cobalt, zinc, chromium, and nickel. These 5 heavy metals were considered, and this was preliminary based on the MIC results. And these are the growth curve, and here much variation actually has been found in growth pattern of in presence of both normal as well as heavy metal stress condition. And during the growth study, it was found that these isolates displayed much more potentiality in cadmium uptake, because in presence of cadmium, the growth curve growth rate was quite high, and hence the ICPMA study was carried out to estimate the uptake capability during lag phase, early lag phase, and exponential phase. And it was found that during the exponential phase, the isolates displayed much more potentiality in cadmium accumulation, and the results has been graphically represented here. And then cumulative metal stress was detected, and as zinc was found to show simultaneous accelerating growth in most of the isolates. Hence, considering zinc as the constant, rest heavy metals were combined, total 8 combinations in these are the double cumulative metal stress: zinc, nickel, nickel from starting from zinc nickel up to zinc chromium in double cumulative metal stress. For that three isolates, the respective MTC, that means maximum tolerable concentrations, I mean have been found here in this chart. And not only that, the three isolates also responded positively in the triple cumulative metal stress, and the MDC values has been represented here. Next, next, these are the slides showing the growth, growth of these three isolates, and in double cumulative metal stress condition. Here, ABC, means RF2, RF3, and RF6, the three isolates that were selected for further study, and ABC in zinc and nickel. Combination. I mean, as per the MDC values that were shown in the previous slides, and these are the zinc cadmium combinations, and these are the untreated control of RF2, 3, and 6. Then after after analyzing the heavy metal tolerance and uptake potentiality, then plant growth promotion ability was also detected through some in vitro screening tests like phosphate solubilization, zinc solubilization, siderophore production, and it was found that one of the isolates showed a prominent response in case of all the experiments regarding that plant growth promotion potential. This is a figure represents the NBRI, phosphate solubilization in NBRIP medium. This is siderophore production. Here, clear halozone is found, and this represents it is positive the response. And this also, but the change of the filter paper, the color change. From yellow to orange, this represents the positive response in HCN production or hydrogen cyanide production. These are the IA production, indole acetic acid, and zinc solubilization. In all the results, the isolate RF2 responded positively. And next, this is the pH growth curve, and pH tolerance of the isolates were also studied, and they responded best from pH 4.5 to 12, and here this is schematically represented, and these are the scanning electron micrograph of the three isolates that were studied, and next, finally, molecular identification revealed the isolates as the strains of, showing the maximum similarity with Pseudomonas aeruginosa DSM 50071, and that belonging to the phylum Proteobacteria, and all these three represents much more potentiality regarding heavy metal tolerance, and one of the isolates showed that plant growth promotion ability also. Okay, so this my conclusion. The heavy metal resistance and plant growth promotion ability, these are the most important aspects regarding the development of eco-friendly, cost-effective strategies regarding bioremediation, and to improve the agricultural productivity and sustainability. This green technology may provide the basis, and this is the pioneering work, and needs to explore more, and further investigation regarding the mechanisms may be carried out, and in vivo experiments may be carried out. This may provide the basis of serving the purpose regarding the sustainable agricultural productivity and sustainability. Thank you.
Okay, thank you.
Thank you, sir.
Thank you, Jayanti, for your nice
Yes.
presentation. Okay,
Yes,
so
sir.
we are running late, shortage of time. Okay, so
Yes, sir.
if anybody have any question, please text on the message box. So later on, she will be answered. But
Yes, sir.
now, Devabrata Modok again joined us.
Thank you, sir.
Okay, thank you. Devabrata, are you there?
Yes, sir.
Okay, you may proceed.
Okay, sir. Thank you, sir. Good evening, respected chairperson, sir, respected teachers, and all our fellow mates. Today I am here to present the seminar topic, and that entitled topic is Drinaria quercifolia rhizomethanoic extract, amylorate, rheumatoid foedema, influence complete adjuvant induced arthritis in rats. And this work has been carried out under the supervision of Professor Shomen Bhattacharya from Department of Zoology, University of North Bengal, and the co-supervision of Dr. Subarna Thakur from the Department of Bioinformatics, University of North Bengal. Um, as we all know, that is the inflammation, that is the body's own defense mechanism. It's a very common biological cascade in any kind of foreign pathogen, tissue injury, or chemical irritation. And it basically acts by removing those injurious stimuli, and it begins the healing process. And this healing process and the inflammatory process, which is very rapid and self-limiting. And both the infectious and non-infectious stimuli can trigger the inflammatory pathway, that is the acute inflammatory response, and that can restore the physiological homeostasis. However, when it is prolonged, it may become chronic, and it enhances the extensive tissue inflammation and contributes to a variety of chronic inflammatory diseases like type 2 diabetes, asthma, ulcerative colitis, or even in rheumatoid arthritis. To treat these inflammatory or the chronic inflammatory consequences, commercially available anti-inflammatory drugs like non-steroidal anti-inflammatory drugs, opioid, even steroids are often used. However, all these drugs have several adverse side effects like drug dependency, hepatic and renal impairment, cardiac arrest, or even cognitive memory deformation, and by taking up the medication for a long time. The scientific community till now has been searching for an efficient medication against inflammation that can minimizes those associated side effects. And in this scenario, the alternative traditional medicine system comes into the consideration, obviously for their pleiotropic nature, due to their easy availability, their low cost, and all obviously for their having lesser side effects. And in this aspect, Drinaria quercifolia or Aglamorpha quercifolia, it is a very popular medicinal plant in this region, and it's commonly known as Oakley farm, and also in Bengali it is called the Pankhiras. It has a great medicinal values against various diseases, and traditionally, the fronds part of this particular plants are used for treatment of typhoid fever, chronic jaundice, headache, cough, cholera, and even skin disease. And the soup prepared from its rhizome is very popular among the tribes of Eastern Ghats, Tamil Nadu region, to get relief from rheumatic pains. And even in the sub-Himalayan region, the tribes like Mech and Koch tribes are used this particular front for the strengthening of broken bones. So my current objective is to validate this particular plant in a chronic anti-inflammatory rat model. So first of all, I have collected the plant specimen, and the plant specimen are identified in the taxonomy department of the University of North Bengal. And from that, there I have prepared the methanolic extract. And and after preparation of methanolic extract, these are the experimental studies. First come to the animal maintenance, and in case of animal maintenance, the oyster albino rats are used, and they are maintained in the animal house of Department of Zoology, and all the experiments are performed under the strict ethical committee, and there is the institutional animal ethical committee permission. First, we have measured the toxicity and the safety dose, that is the 50 value determination, and these tests are done according to the OECD guideline, that is the Organization for Economic Cooperation and Development guidelines. And based on this test, we have selected the doses for in vivo studies, further in vivo studies. And now the in vitro, the in vivo chronic anti-arthritic potentials of this particular rhizome extracts. Friends complete adjuvant, that is the FCA-induced chronic arthritis model has been established as per the standard protocol. And this is the my experimental design for FCA-induced chronic anti-inflammatory arthritis model. And for this particular model, we have considered 4 different groups, and the group 1 signifies the FCA control group, that is the arthritic control group. Group 2 signifies the low dose group of Dynaria methanolic rhizome extract, and it has get the 250 mg per kg body weight of the Dynaria methanolic rhizome extract. And group 3 considered high dose group of DME, and it was received the 500 mg per kg body weight of the DME, that is the methanolic extract of the particular plant species. And group 4 was considered as the vehicle control group. And except this group 4, all the others group animal have received an intradermal injection of 0.1 ml of FCA into their right hind paw of their subplantar region, and on day 0. And on day 14, they have also received a booster dose of the same amount of FCA into their particular right hind paw region. And except the vehicle group and the arthritic group, the low dose group and high dose group continued the Dynaria methanolic rhizome extract treatment from day 0 to day 28. And after the day 28, all the animals are euthanized, sacrificed, and after that, the blood was collected from cardiac puncture, and different tissues are harvested, and they are further analyzed for different parameters. Now come to the result section. This is the result of the acute toxicity test, LD50 value, and the determination of oral feeding dose for further studies. And in this particular group, we have chosen 4 group, and the highest dose was taken, that is the 2000 mg per kg of body weight. And up to this dose, there was no behavioral abnormality was seen in any group, any animal for any group. And the normal body weight gaining was observed in this particular bar diagram up to the 14 days of observation. And based on this result, the lethality value of Dynaria methanolic rhizome extract, that is the DME, was considered unclassified up to the feeding range. And based on this result, we have choose the low dose group, that is the 250 mg per kg body weight, and the high dose group, that is the 500 mg per kg body weight for further in vivo studies. This is the result of in vivo anti-rheumatoid activity of Dynaria quercifolia. In this particular figure represents the rat paw edema expressed in circumference in different experimental groups. As in this particular figure, we have seen that in the day 0, in the arthritic control group received the FCA like the other dose group, but the from day 0 to day 20, the arthritic control group arthritic control group having showed the highest amount of paw circumference. And treatment with Dynaria methanolic extract in both the low dose group and high dose group significantly ameliorated the paw edema during the experimental schedule when they are compared with the that arthritic control group. And this is the data that represent the mean and ACM value of 6 animals, and we perform a 2-way ANOVA followed by the direct multiple comparison test to statistically validate this result. Now come to the hematological parameters. In the figure A represent the hemoglobin concentration of different dose groups. As we all know that in case of arthritis condition, the hemoglobin concentration became low as followed by the RBC concentration. But treatment with the low dose or high dose group, that is the treatment with Dynaria, significantly normalized the hemoglobin content as well as the RBC content in the experimental groups when we compared them with the arthritic control group. And in case of WBC and platelet, as we all know that that for inflammation there was a significant amount of WBC present in inflammatory condition, as that is seen in also in our FCA-induced arthritic model. But treatment with diarrhea significantly decreased the elevated amount of WBC in both the low dose and high dose group, and also same result was observed in case of platelet group when we compare these stroke groups with the that of arthritic control group. And here in this particular slide, we have represented the data as the mean and standard deviation, and they were analyzed using ANOVA, one-way ANOVA, followed by the Dunnett post-hoc test. This slide represents the biochemical parameters of different experimental groups as part of the treatment schedule. Um, as we in particular first one, that is the in total protein concentration, as we all know that in arthritic concentration the total protein and albumin both are getting low in the disease condition. But treatment with diarrhea methanolic extract, we have seen that in both the low dose group and high dose group, the total protein concentration were nearly normalized. However, in case of albumin, we have seen that only the high dose group significantly normalized the albumin level when both are compared with the standard group, that is the arthritic control group. And in case of creatinine group, we have found no alteration, no significant alteration in any kind of group. Yeah, and these represent the normal renal function. And in case of ceruloplasmin, this is acute inflammatory marker protein, and this is very important, and it has got its number increased when there is an inflammatory condition. And but when we treated our experimental groups treated with the low dose group and high dose group of DME, we have seen that the DME can normalize the elevated serum ceruloplasmin level and significantly when we compared with the standard when we compare the arthritic control group. These are the this picture shows the radiological analysis of the right hind paw ankle joint of experimental rat groups. Here AE shows the vehicle group, BF shows the low dose group, CG shows the high dose group, and DH shows the arthritic control group. And as we see, this arrow indicates the swelling and the degradation of cartilage in the experimental animals. And in case of arthritis control group, it has been shown that the swelling and the degradation of cartilage was severe than the normal group. And when we considered our low dose treated group and high dose treated group, in both cases we have seen the treatment with DME nearly normalized the treatment with DME inhibit the bone degradation and swelling in both the experimental groups. And this figure represents the histological section of rat paw ankle joints obtained from different experimental groups. In this particular figure, A represent the vehicle control group, B and C represent both the low and high dose group respectively, and D represent the arthritic control group. And here white arrows represent the destruction of cartilage lining, and black arrow represents the immunocell infiltration in throughout the slide. And as we seen that in case of arthritic control group, the cartilage destruction was prominent and the immunocell infiltration were higher, but a normal histological architecture was observed in case of the vehicle control group. However,
Try to conclude.
However, when we observe in the low dose group and high dose group, there is a reduction in the infiltration of white immunocells, and also the normal joint space, that is the normal cartilage lining was observed. And this is the histological section of kidney, that is the upper panel, and the lower panel represent the histological section of liver. And no alteration of kidney and liver section are observed, and this signifies there was no significant toxicological parameters were observed in up to the 28 days of experimental schedule. Now come to the discussion. Dineria methanolic, Dineria quercifolia rhizomethanoic extract, that is the DME, was considered non-toxic up to a dose of 2000 mg per kg of body weight. And DME attenuated the inflammatory poedema in both low dose and high dose group when compared to the arthritic control group. DME significantly increases the RBC and hemoglobin counts and can also decreases the elevated WBC and platelet counts in both the low and high dose treated group, and thus it reflecting its immunomodulatory effect. DME significantly increases the total protein level and albumin level and decreases the elevated ceruloplasmin level, reflecting its tissue repair effects. And from the conventional radiographs, we have seen that treatment with DME prevented bone joint destruction, and this was also further supported by histopathological analysis of ankle joint, ankle joint, and no alteration was observed in the serum creatinine level, who is further supported by histological studies of liver and kidney section that exhibiting any sign of hepatotoxicity and nephrotoxicity. These are the references that I have mentioned in this particular slide. Thank you.
Okay, thank you, Davabodo, for your nice presentation. So now I would like to invite Ravi Prasad for his presentation. Ravi, are you there?
Yes, sir, I am here. So just
Please,
I am
please.
sharing my screen. Hello, am I audible now?
Yes, yes.
Yeah.
Proceed.
Well, again, good afternoon to all of you. Myself, Ravi Prasad, and today I'm going to talk on an important topic, that is the important medicinal plant of Sariska Tiger Reserve, Rajasthan. Whenever we talk about the desert state of India, that is Rajasthan, our general perception is that the state is having only a thorny and prickly species, but actual scenario is not like that. So today I'm going to take you to the journey of the medicinal plant of Sariska, how the nature has blessed this desert state with a number of medicinal plants. This is my study area, Sariska Tiger Reserve. It is situated in the Alwar district of Rajasthan, which is in the northeastern part of the Rajasthan, and the total geographical area of the Sariska is 1213 square kilometer. The biodiversity of Sariska is quite rich. In some part, it is having a scrub forest. The vegetation of the Sariska corresponds to the northern tropical thorn forest and northern tropical dry deciduous forest, and at the same time it is having a very good desert element, desert vegetation and wetland like situation. So it makes the biodiversity of the area unique, and as a result it harbors a number of medicinal plants. And some of the very important medicinal plant of the region are the Abrus precatorius, although the seeds are quite poisonous, but after several rounds of boiling, people are using to treat for different kind of heart diseases, heart ailments. And this is the datura metal, and this one is the asparagus racemosus, that is quite common in the region and highly used by the local people. And this is the Cisampelus perita, that is also a very important medicinal plant in the desert region. And this is your Mucuna pruriens, and here I am learning some of aspects of the medicinal plants from the local people. And this is the your Cocculus pendulus, and this is another important plant. This is, hello, am I audible now? Hello. Yes, sir. And this is very important, another plant, that is the Tribulus rajasthanensis, and its fruits are used to boost stamina, and this is another important element of the desert. Just a second. This is Citrullus colosensis, that is widely used to for as a multivitamin supplement by the local somnifera. The root of Gloriosa superba is used by the local people to as a anti claimed to have anti-cancerous properties. And some of the shrub species, the area is having a very good population of your Barleria prionoides, that is locally known as the Bajradanti, that is used to cure the different toothache problem. And this is another species that is highly medicinal, that is the Barleria prionatis variety dicantha, this pale yellow one. And this one is your Caparis decidua, and the fruit is known as the desert apple. This is again highly medicinal plant. And this is your Dichroistrachys cinerea. Its flower are highly medicinal and possess number of antioxidant property. And this one is your Grevia tilifolia, Securenga leucopyrus, and this is your Gigifus numularia. The root of the Gigifus numularia is used for different kind of the backbone related problems. And this one is your Silvadora persica. You might have seen the advertisement of the Miswak toothpaste, and the major constituents of that toothpaste is this Silvadora persica. And this is your Grevia tenax. Grevia tenax is very important element of the desert. And in whole Aravali, you will find one species, that is the center one. This is your Euphorbia caducifolia. Its stem are highly medicinal and used by the local people for treating number of different ailments. And this is your Vitex nigando, the pale blue flower. And the leaves of these plants are boiled, and after boiling, it is used to treat the viral fever. And this is another important species of the desert. This is your Acacia jacqueminmonsi. But due to over exploitation of this plant, now it is quite, the number of this species reduced in the wild. And this is another important species, Celastrus paniculatus, locally known as the Malkangni, and it is well described in Ayurveda, and at the same time local people are still using it. And this is some of the important tree species. Our general concept is that the desert is having a very less number of tree species, but in Sariska it is having a very good population of tree. This is your Acacia nilotica, Babul, Casseria. This one is your Casseria tomentosa. This is again highly medicinal. And we are very familiar with the moringa, but this is the another species of the moringa that is endemic to the desert area. This is your Moringa concanensis, that is having a anti-diabetic property. And this is another important plant. This is Boswellia serrata, and its gum is highly used, is having a number of medicinal property. And this your pale orange to pale yellow flower, this is the state flower of Rajasthan. This is Tecomella undulata. It is having a number of medicinal properties. And this below one, the pale yellow flower and the pod, this is the state tree of the Rajasthan. This is Prosopis cinerea, and its pods are highly medicinal. It is used to boost stamina, and at the same time it cured the number of stomach related problem. And this is your Rhytia tinctoria. The latex of this plant is highly medicinal, and this is a very common element, although it is also found in the Himalaya and even in Siliguri region. This is Cassia fistula. It is quite common. And the whole Aravalli region, the most dominant tree is your Anogesus pendula, and Anogesus pendula is highly medicinal. Its stem is used to treat different, having number of medicinal property. And this one is your Balanitis aegyptica, and its fruits is highly medicinal. And the whole valley of the Sariska is having a very good population of the Butea monosperma, that is again highly medicinal plant. And if I will summarize the synopsis of the medicinal plant in Sariska Tiger Reserve, then I can say that it is having a more than 96 species of the herbs, more than 35 species of the shrubs, 9 climber species, and 17 tree species, which are having a number of medicinal property. Altogether, till my survey, I have their number accounts near around 157 medicinal plant, although the number will increase because the survey is still going on. And here I have noticed that the area is having very good population of the Justicia adaptora, which is in Bangla, it is locally commonly known as the Bashok Pata. In Rajasthan, it is known as the Arusta or Arusha, which is widely described in Ayurveda and used to treat the different cold and cough related problems. The but the but unfortunate situation is that the population is quite luxuriant in the area, but not the government, not the forest department is taking initiative to set an industry for the medicinal plant. If they can take the initiative, they can set up the medicinal plant for such widely growing species like Justicia adatora in the region. And nature, if you use the plant given by the nature in a sustainable way, then we will get everything from the nature. But due to the greed of the human being, as you can see, this is the bark of the Terminalia arjuna. But what people are doing, they are continuously removing the external bark. As a result, the life of the plant get reduced and the multiplication rate get reduced. And this is the Butea monosperma. To 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 extract the gum from this plant, they have made a number of cuts, and you can see the number of cuts on the stem of the plant. It simply reduces the lifespan of the plant, and as a result, they become reduced day by day. And I have performed one special study on one important medicinal plant. This is a critically endangered medicinal plant. This is Commiphora whitei, that is commonly known as the Google. It's a and its oleoresin is highly medicinal, which is having a medicinal property like the expectorant, carminative, aphrodisiac, demulcent, anti-inflammatory properties, and at the same time it is used for the rheumatoid arthritis, osteoarthritis, obesity, and several other disorders. It lowers the cholesterol level of the blood, and it is also having a Google cholesterol 1, 2, and 3, Google lipid, gum resin. But what people are doing nowadays, what greedy people are doing, they are adding copper sulfate to extract more and more oleoresin, and as a result, its population is quite reduced, and now it is under the category of the critically endangered. And one more problem with this species is that it grows in a special physiological condition and it needs a phytosociological condition, but its phytosociological condition in the natural habitat is totally disturbed, as everywhere in the Rajasthan, in Aravalli, mining is going on. After 2 to 3 years of age, the plant produces number of seed, but the germination power of the seed is very poor, as the plant grow in a special physiological phytosociological condition, but this phytosociological condition is its habitat is totally disturbed. It grows with the species like the Grevia, Acacia, Ceropegia, other Euphorbia species, and it also provides the shelter to the CITES species like the Ceropegia, this one, Ceropegia bulbosa. But due to the habitat destruction and the disturbance in the phytosociological condition, the population is reduced to a great extent, and I have tried to multiply with the conventional method following the ex situ conservation. Here I have taken the stem cut of the 20 to 25 centimeter into 1.5 into 2 centimeter stem cut, and after cutting it, I have soaked in an IBA solution, indole 3 butyric acid, 5000 ppm, following the quick dip method, and then it was transferred in a moist soil, having the composition of the sandy and gravelly soil into 2:3 ratio, and then it was transferred into the pot, and the new leaves and root initiation take place after 10 to 15 days, and the new plant list is ready for shifting into the another large pot after 60 to 75 days, or after almost 3 months you can shift it to the field. Here it is the almost 3-month-old plant, and then I have transferred it to the as a ex situ mechanism. I have transferred it to the field, and after the growth of 1 year, it has started giving the fruit, and this is the different growth stage. And this cutting method has given a very good result, and germination percentage in this cutting method was 60 to 70%, and in case of the seed germination percentage was 10 to 20%. But the one important thing is that the health and vigor of the plant obtained from the seed is a very healthy one. And finally, from the study of the area and the medicinal plant of the area, I can conclude that the local people and the medicinal practitioner, where they collect and use the medicinal plant in the crude form, but their traditional knowledge are ever depleting due to the encouragement of the modern medicine in everyone's life. So their accurate documentation is urgently needed coupled with the scientific research-based evidences. Sustainable use of the medicinal plant under the strict preview of law need to be ensured with major awareness campaigns among the locals for conservation of such medicinal plants. Local people can be promoted and encouraged for access to conservation of medicinal plant. There is urgent need to check the grazing outside the forest. The whole district of Alwar is famous for its milk egg. That means naturally it is having a very good population of cattle, and there is illegal migration of the domestic cattle in the century. They are destroying the number of plants, and at the same time we have to stop the entrance of the number of weed species. They are also a big threat for such medicinal plants. And if we can stop, as in the first picture you can see, this one person has almost chopped the tree of the Anogenesis pendula, the most dominant tree. But if we cannot stop such activity, and this one is the widespread population of the Cassia tora, it is almost destroying everywhere. It is covering the
Ravi, you are not audible. Okay, Ravi almost conclude his presentation. So thank you, Ravi. Thank you for your informative talk about the arid flora and their medicinal use from the uh, Rajasthan Sariska Tiger Reserve. Thank you. Now I would like to request Dr. Sudeep Kumar Roy to present his talk on effects of habitat fragmentation on the reproductive success of Almas wallichiana, plancho, almisi, a rare and threatened tree species of Central and Western Himalaya.
Yes, sir. Good evening, all.
Please. Good evening.
Good evening, sir. Presentation is. Can everyone see the presentation?
Yes.
Yes. Okay. Good afternoon, everyone. Organizers, my colleagues, other participants. Today, my topic of presentation is the effect of habitat fragmentation on the reproductive success of Almas wallichiana, rare and threatened tree species of Central and Western Himalaya. So first of all, I would like to start with the biodiversity and the number of species we have in Himalayas, like 8,000 angiosperm species, gymnosperm, 600 pteridophytes, 1,737 bryophytes, lichen species 11, more than 1,000 lichen species. So it is one of the most biodiversity hotspot of the world. So definitely, and it also faces a high threat to its biodiversity due to land use change, deforestation, infrastructural development, and climate change. So this land use change, deforestation, and infrastructure development has resulted in the habitat fragmentation of many native tree species. One of the major consequences of decreased densities of a plant in a population is the reduced probability of successful pollen transfer due to increasing distance between the individual plants. And this results in the pollen being often wasted due to failure to reach conspecific stigma and a subsequent reduction in quality of pollen, total pollen pool. So this we can all conclude that this reproductive failure is penultimate to species extinction, and this is why the detailed study of reproductive biology of following plants or that will provide the basic and crucial information on the ecological constraints and by the species in nature. And it is also imperative that studies on this reproductive biology should be effectively accomplished for development of an effective management plan for threatened and rare species. And understanding the pollination biology and other typical breeding system of the plants, this reproductive behavior of a plant species are vital for predicting the consequences of human disturbances in the forested ecosystem. So tropical countries like India, we lack baseline data on reproductive biology or ecology of this species, and studies on the reproductive biology should be effectively accomplished for strategizing any successful conservation plan for the species. So I will be presenting this slide just to show that there are there are very we face critical challenges during this reproductive field biology study and during at the field, because the physical location of the trees are sometimes it's very hard to reach, and to find a suitable population for experimental setup where we can exactly study its flowering period and the fruiting period. So that suitable kind of population and accessing it, the flowers, the exactly the Himalayan species, they have this synchronous and brief flowering period, and the distance between two populations, it makes very hard to exactly study the reproductive biology of any species in the nature. So and nothing to sort that natural calamities and other things are also there, then field to laboratory transfer specimens in right condition. So now coming to the we have already discussed the importance of reproductive biology in conservation of threatened tree species. This present work was undertaken to study the reproductive ecology of rare and threatened tree species, like this is Ulmus wallichiana, and it is belong to family Ulmaceae, known as Himalayan elm. And the absence of new plantation and unabated lopping, these Ulmus wallichiana have become this rare and threatened in India, according to ICN Red List as well. This is also vulnerable, falls under vulnerable category. So this present study we had taken up for to recommend effective species-specific conservation and restoration measures, and to look into to specific objectives, like to investigate the pollination ecology of Ulmus wallichiana, examine the degree of pollen limitation, then determine its breeding system. And understanding of these aspects, we know the critical for conservation of Ulmus wallichiana in natural conditions. So basically, the genus Ulmus, it consists of a deciduous and semi-deciduous woody plants distributed in northern hemisphere, including North America and large part of Eurasia, and then in our on Asia continent, this Himalayan elm, the tree species found in mountain zones of central western Himalaya from Afghanistan to Pakistan, northern India till Nepal. And within India, we can find at Jammu and Kashmir, then Uttarakhand, Himachal Pradesh at the height of 1800 to 3000 meters. So coming next to the main problem. So this is the lopping and the yeah. So we have studied we have accessed the populations. So, on the basis of available literature on the natural distribution of Ulmus wallichiana, we surveyed, we done some field surveys, and then we carried out, we went to different floras and different herbariums. Then we located this in Dehradun district, this Jadi and Konen village, and with we specifically taken some populations which was present naturally there. So coming to the flowering floral architecture and of Ulmus wallichiana, it showed that it is exclusively wind pollinated species. The flower lack nectar and were not observed to be visited by any insect. And morphological features were also conductive to wind pollination. So next we coming to the breeding system analysis. So Ulmus wallichiana is mostly a xenogamous species with a small proportion of fruit set through selfing. So observe, it is primarily showed that through selfing, there was a very small proportion of fruit set. Then, however, natural population of the tree species are small, with low density, and distances between trees are large for effective pollination by wind. So, resulting pollination constraints lowers the natural fruit set as species experience significant pollen limitation. So selfing does not appear to be assuring the reproductive limitation, rather lead to inbreeding depression, which manifested in forms of seed abortion. Then coming to the pollen dispersal part, the pollen dispersed through wind showed a leptocartic distribution, and the density of airborne pollen grains declined with the distance from the source plant, and it was highest in the canopy region, so of the source tree, and it declined to almost 10 to near zero pollen count was there after 50 meters distance from the source plant. So no pollen grains observed beyond 50 meters, and wind pollinated species, the effective distance of pollen flow is influenced by a lot of uh, characters like densities of conspecifics, topography, and intervening vegetation and wind speed and direction. So this reduced pollen transfer eventually has detrimental consequences of production of new individual and sustainability of a population. So this species also we have established by pollen supplementation experiment that pollen supplementation of this plant show an increase in the percent fruit set compared to controls, and open pollination yielded average 24%. Then when supplemented from the same population and other population, the yield was, the fruit set was higher, like 53 and 56. And the present study provides experimental evidence that this pollen limitation within population of Almus wileychiana, of those population we have studied, and pollen supplementation experiment resulted in higher fruit set. So it may be inferred that reproduction limitation is limited due to pollen limitation of the species, and pollen limitation may not commonly found in wind pollinated plants, but density of specific and pollen transport distances may perhaps have a bearing on success of this species, Almus wallichiana. So after we can conclude and some recommendation for the future this conservation of this species, that due to unabated lopping and natural recruitment process of Almus wallichiana, natural recruitments and numbers has declined, and this present study has provided vital information on reproductive strategy of Himalayan elm in its natural habitat. And since it's primarily xenogamous species and exhibits a small proportion of fruit set to selfing, and natural population of these tree species are very small and low density, with the distances between trees are larger for effective pollination by wind, resulting the pollination controlling lowers natural fruit set as the species experiences significant pollen limitations. And this also probably indicates the fruit production is not only limited by pollen quality but also by quantity, but also pollen quality. That pollen dispersal between nearest neighborhoods than across a population reinforce local spatial genetic structure and genetic neighborhoods. Then selfing does not appear to be assuaging the reproductive limitation, rather leads to inbreeding depression, which is manifested in the form of seed abortion, resulting in empty summers. So in view of all these findings, we can recommend that reintroduction of the saplings at the site of other site with distances between plants not more than 30 to 50 meters may be considered for improved outcrossing opportunities and for larger number of natural and new recruits. So that will be all for today's topic. I would like to thank University of Delhi, and UGC and Ministry of Environment, Forest and Climate Change for during my research. I got fund for this specific study for reproductive biology of rare, endangered, and threatened species. And my supervisor, Professor A.K. Bhatnagar and Professor A.K. Pandey, they have guided me through this research. Thank you very much, and that will be all.
Okay, thank you, Dr. Roy, for your such informative talk and in-depth research on the conservation and the reproductive biology of the rare and threatened species of Almas wallichiana from central and western Himalaya. Thank you
Thank
very
you,
much
sir.
for
Thank you
your
very much, sir.
nice presentation. Thank
Thank
you.
you very much, sir.
Now
Thank you.
I would like to invite Ume Begum, and her title of presentation is Endemic Plant Species of Northeast India. Are you there, ma'am?
Yes, sir.
Yeah. Please, you may start your presentation.
Yes, sir. Good evening, everyone. Myself, Ummi Tawhida Begum, pursuing BSc in Botany, Department of Botany, Maryam Azman Women's College of Science and Technology, Hudai. Today I am going to discuss about the endemic plant species of Northeast India. Endemism, the endemic species, the characters and the factors responsible for the endemism and what are the causes of endemism, and then the what are the phytogeography of Northeast India, and then the endemic species in the India, Northeast India. Later we will see the strategic goals for the conservation and then the conclusion. So now the meaning of conservation. What is endemism? Endemism means the confinement of a particular species, genus, or group of animals to a particular area. Taxa found only in the single restricted geographical area is known as the endemics. Endemism normally applies only when there is a considerable restriction in the area of distribution. Endemism is the ecological state of being endemic to a different geographical location, such as island, nation, country, or other defined zone or habitat types. The opposite of endemism is called the cosmopolitan distribution. Now, the endemic species. The concept of endemism was first given by the Kendall when a species is found only in a particular geographical region because of its isolation, soil, and climate condition. It is said to be endemic. Endemism is basically of the following types: the new endemism, paleoendemism, and the epibiotic or relic endemism. The number A is the neo-endemism. Neo-endemism is a taxon of evolutionary young and not yet spread over the new area. Example, Senecio cambrensis. And the paleoendemism, the taxon is restricted now, but once it was widely distributed. The restricted of the species is a pocket is due to the physical barrier like the desert, mountain, sea, etc., or change in climate or soil type, etc. And the epibiotics or the relic endemics. The plant belongs to the fossil group and are restricted to a few pockets due to the favorable climates, lack of competition. Example, Ginkgo biloba, which is restricted to China but widely spread in the north temperate zone as a fossil. According to Richardson in 1978, endemic is an intermediate between the two extremes, that is, plants which are not of the recent origin but have retained a narrow distribution, and he called the holoendemism. If the local condition induced reactivation of the paleoendemism, evolving new endemic species after a long gap, they are called the active epibiotics. The degree of isolation of the area evolved is usually proportional to the percentage of endemic species in flora. The degree of isolation is measured either as the distance from the similar areas or the length of the time that the area has been isolated. Now, the next one is the endemism represents a unique step in the process of evolution which caused by the perpetuated and sustained only in the locality concerned depending on the environmental quality. The importance of the habitat is much as is the most of the cases such localities possess a number of endemic species distributed in several taxonomic categories. Endemism are sometimes restricted to a very small area. They are termed as the local endemics. Endemics arising due to the mutation are called the pseudo-endemics. Example, Franklinia elotamaha. Only one plant of this family was present in the 18th century of Elotamaha River in Georgia. Larger area usually have a greater proportion of the endemics than the smaller area. If the whole world is considered as a one digit person, then the number of species endemic to the Eastern Alps or to Western Alps added together amount of 78% of the total number of Alphic endemics. Endemism refers to the species that was formerly widespread but is now restricted to a smaller area. But on the other hand, new endemism refers to the species that has recently arisen, such as species that has diverse and become productively isolated, or one that has the form following the hybridization and is now classified as the separate species. Protecting just 1.4% of the world's land surface containing all the endemism hotspot, 44% of the world's vascular plants, and 35% of its terrestrial vertebrates could be preserved. But these hotspots also contain the 20% of the world's population. Most of them
rapidly increase and are therefore areas of intense commercial exploitation and habitat destruction. Therefore, therefore, the successful conservation cannot focus on wilderness areas alone. A lot of work is going on the new endemism. On the basis of cytotaxonomic studies, Favor and Contrario Perez in 1961 differentiated three types of new endemics: schizoendemics, petroendemics, and apinoendemics. Schizoendemics derive from the having given rise to a more widespread taxon of same chromosome number. Petroendemics restricted diploid which have given rise to widespread polyploids. Apoendemics is restricted polyploids which have arisen from the widespread diploids. There is a great confusion in the term endemic, rare, relics, etc. All endemics are not relics as there are a large number of new endemics. All endemics are not rare as some are abundantly present in the particular locality. All rare plants are not endemics. Some may occur at the several places with few representatives. Now, the character of endemism. They are localized in the distribution because of their narrow ecological amplitude and are unable to invade in the fresh areas. They lack potentially to migrate because of saturated genome. Real endemics never migrate while the new endemics have the potential to migrate. The dispersal propagules are not able to sustain during migration to other area. It may be due to the physical barrier. Then the theories of the endemism. The first theory believes that the last survivor of the flourishing flora which is now declining are the relics or epibiotics, which are endemic. Second theory believes that these are recent and useful forms in the course of gradual extinction. This theory is also known as the age and area hypothesis. Factor affecting for endemism. Factors responsible for the production of endemism are naturally crossing among the closely related plants, growing under favorable conditions and mutation. If the condition of isolation is developed by the effect becomes more pronounced. Endemism is found in example island, isolated areas, etc. Mountains also have the more endemic species as they are isolated. Example, 70% species of Himalaya is endemic. Climate Climate also is one of the factors. Example, Northeast India display tropical climate, monsoon climate, climatic northeast between the valleys and the mountain region. Now, the list of the endemic floral species is the Nepenthes khasiana. It is carnivorous plant endemic to the Khashi Hills of the Meghalaya. It is critically endangered also. This is called the pitcher plant. And this Eventus mimosa is from the family Acanthaceae. Azeptis congelali. Idiocracy. And the Magnolia bellioni is from the family Magnoliaceae, and the Discaria albifera is from family Scaliaceae. Then the citrus appearances family Tiaceae, then here the Hymenacea from the family Poaceae, Selexia Gentiaceae from the Clustraceae. Here, the endemic taxa of the Manipur is here the list of endemic species found in the Manipurs, and here all the endemic taxa found in Meghalaya. These also are from the Meghalaya, and the endemic taxa of the Mizoram, like the Begonia vivacensis, there is Lea heresia, Benjari gesens USA. The
और भी है भी
endemic taxa of
5
Nagaland from the Begonia vati, Risalisa vati, there, and here the endemic taxa of Sikkim, Hemon Demesia hook, Erenia Tengiacenesis. The endemic taxa of the Arunachal Pradesh area, the like the Citria macrophyllaceae, Chias achamentia. The endemic taxa of Assam are here, the Acacia dardenia, Ardisa paradilum, etc. And we now come to the strategic goals for the conservation and suitable use of biodiversity. The conservation of biodiversity through the national parks and wildlife sanctuary. Protection of indigenous species of the plant and animals against exotic species. Encourage the research scholars
Okay,
to
madam,
study
please
the use.
conclude. Try to wind up, please.
Yes, sir. Encourage the research scholars to study various aspects of the biodiversity in nature, etc. Awareness amongst the people regarding the importance of biodiversity and nature. Here the conclusion, the Northeast region is blessed with a variety of rare and endemic plants, animals, and microorganisms. The mountain forests and rivers provide this region with best of climate to nurture this flora and fauna. The awareness is the most important key to conservation and continuation of this endemic species. Northeast India is a biodiversity hotspot in enriching the country with the most colorful and fascinating species. So here the reference from which I got all the information about the endemism. Thank you.
Okay, thank you, madam, for your presentation on endemic species, their categories, and the species of various states of Northeast of India. Thank you very much for your such a nice talk. Thank you. Now I would
Thank
like
you.
to invite you, Nikki L. Kungate, and her title of topic is Phytoliths of Bamboos in Barak Valley, Assam, a study.
Thank you, sir.
Please,
Allow me to share my presentation.
please, please.
Okay, sir, is my screen visible?
Yes, visible. Continue.
Okay, thank you, sir. Okay, so a very good afternoon, sorry, evening to the organizers, respected teachers, and all fellow participants. So my name is Uniki L. Kungate from Assam University, Silchar. So today I will be presenting on the topic phytoliths of bamboos in Barak Valley, Assam, a study. This is a part of an ongoing research under the supervision of Dr. Devjyoti Bhattacharya, Assistant Professor, Department of Life Science and Bioinformatics, Assam University, Silchar. So phytoliths, as we all know, are minute particles of silica, and they are formed within most plants after they get absorbed by the plant cells in the form of monosilicic acid. And these phytoliths are of distinctive and repeatable shapes, and they remain intact in most soils and sediments for a very very long time, and these phytoliths are very much microscopic in their size, ranging between 10 to 70 microns. And the use of the term phytolith is credited to Ehrenberg, a German natural historian, and it is derived from a Greek word which means plant stone. And Ehrenberg published his illustrations of the phytoliths in his seminal textbook Microgeology in the year 1854. So the plant silica in particular are opaline in nature, and they are found in the internal tissues, but mostly they are found in the epidermal layers or the epidermal tissues. And the nature of the plant silica and the distribution of silica bodies in different plant families are very much well studied and documented by researchers like Simpson and Volcani, Ollendorf, and his co-workers, and other researchers as well. And phytoliths have also been isolated from atmospheric dust, soil, ancient sediments, and deep sea cores. And phytoliths from different plant taxa tend to possess characteristic morphological features. Hence, several investigators have attempted to identify the plants from which these phytoliths may have been derived. And phytoliths of grasses are of utmost interest in this respect because all members of the Poaceae family accumulate large amount of these silica bodies. These are some of the types of the silica morphotypes that are found in the groups of grasses. Now, talking about the taxonomic role of phytoliths in identification of bambuaceae. So bamboos are a group of perennial giant woody grasses belonging to the family Poaceae and the subfamily Bambusoideae. And the tribe Bambuaceae includes approximately 60 to 70 genera and over 1,500 species in the world. And in India, these bamboos are represented by around 148 species under 29 genera. And these bamboos are very very much important economically. The we These bamboos found their uses as food, as construction material, and a lot of other uses. So for it being such an important uh, economical plant, the proper identification of the group of plants is very very much essential. And traditionally, bamboos, like all other plants, their identification is based on the morphological features like the rhizomes, the buds, the leaf, the branching patterns, etc. And bamboos are also known to possess exceptional features like the comb sheet and the well developed branching, which are in fact absent in other groups of grasses. And thus these features plays a very important role in the identification and systematic grouping of the bamboos. But these characters, although very much useful, are not very much uniformly applicable to all the species of the tribe Bambuaceae. And thus there is a very absolute necessity to have detailed descriptors which can be followed to identify the bamboos at species level. Now, talking about the identification of bamboos again. So the reproductive characters are the main tool for conventional method of identification of any species of plants, and this has been problematic, especially in case of the bamboos, since these reproductive characters are very much rare, or we can say not readily available, due to their similparous and irregular flowering nature. And thus there is an urgent need for alternative methods for taxonomic identification of bamboos. And in this case, phytolith characters from the leaf epidermis, or maybe from the comb sheet, may serve as an important identifying feature in absence of the reproductive characters. Now, talking about some of the pioneer researches that have been done in the grass phytolith as well as the bamboo phytolith. So the work of Thomason, uh, uh, who used the silica cell patterns in epidermal leaf fragments, as well as other epidermal characters to classify Miocene grasses, is one of the most pioneer work in this aspect. And talking about the Indian scenario, the work by Krishnan et al. In the year 2000, in which they have provided an identification key for grasses purely based on the shape and sizes of the phytoliths. And also uh, Again, in the Indian context, NASCAR and BERA in the year 2018 have also used this the assessment of the opal phytoliths from grasses of the deltaic West Bengal, India. And in this study, they have concluded that the grass phytolith, provide useful and significant information for distinguishing the grass taxa. Now, talking about the bamboo phytoliths in particular, the research by Gu et al. In the year 2006, in which they have used comparative analysis of the cuneiform bulliform cells and the oblong concave saddles, and found that the Dendrocalamus has a separate partition from the Bambusa and Calamus, which might provide strong evidences that Dendrocalamus should be an independent genus within Bambusoideae. And also the research by Tao et al. In the year 2020, which is fairly very much recent, they have showed that morphometric and assemblage analysis of phytoliths was useful in distinguishing 17 woody bamboo species within the genera the Phylostachys, the Dendrocalamus, and Bambusa in subtropical southwest China. Now, talking about the research gaps, the significance of phytolith characters in grass taxonomy as a whole have been found useful by many researchers. And although morphotaxonomical study on bamboos from the study area, that is from Barak Valley, have been conducted by Devi et al. In 2017, wherein they have reported 19 species under 6 genera. But till date, there is, the study on phytoliths of grasses and bamboos in particular have not been conducted in the northeastern states of India. Hence, an attempt is made through this research to fill the lacuna in the field from Barak Valley region of Assam. So for this particular study, three objectives were taken up. The first one is a study of phytoliths from both fresh as well as herborized materials, systematic analysis of the experimental data, and identification of taxa on the basis of the phytolith features. Now, moving on to the methods and methodology. So, like mentioned in the objective, so the present study have utilized the fresh as well as herborized material available from the study area. Uh, the study area the study is based in the Barak Valley region in the southern part of Assam, and the region occupies an area of 6,922 square kilometers. And the Barak Valley consists of three districts, namely the Kachar, Karimganj, and Hailakandi. And the valley shares its boundaries with the Mahasau district and Meghalaya in the north, Manipur in the east, and Mizoram in the south, Tripura and Sylhet district of Bangladesh in the west. And the vegetation type of this region is mostly represented by tropical evergreen and semi-evergreen types. So the map of the study area have been provided. So now talking about the preparation of specimen for the epidermal studies. So the leaf specimens, both dry and in fresh form, after cutting into segments, are subjected to
Uniki,
treatment.
you show your result and conclusion, please. Okay.
Okay, sir. Okay, so like I've already mentioned, this is an ongoing research. So till now, the data that we have available are from the epidermal studies. So for the epidermal studies, 17 species of the bamboos have been done. And in these, what we have observed is that all of the species of bamboos under all of the 17 species had the uniform shape, that is the saddle shaped phytoliths were very much uniform in these species, kind of showing that these saddle shaped phytolith is a diagnostic feature of the tribe Bambusi. And the size of the phytoliths showed some variations in the in the different species and different genus. And with proper magnification and isolation of the silica bodies and proper measurement, these can be helpful in delimitation at the genus and species level as well. So the same thing has been explained here. So these are the photo plates from of the epidermal peels. So here 8 slides have been shown. So if I zoom out, so These are the phytolith structures that can be seen, and these are arranged in certain rows. And these slides, these photographs were taken from the coastal region of the epidermal peel slide that were prepared. Okay, now coming to the conclusion, this study reveals that the phytolith characters, especially the shape and the size from the epidermal tissues of bamboo leaf, have significant values in the classification at tribe and in some cases genus level as well. And proper study, that is proper analysis of the individual silica bodies, will be helpful in delimitation at genus as well as species level. And phytolith relevance in systematics of bamboos, although an important area, is still not adequately studied. And morphotaxonomical studies have been the basis of identification and classification of bamboos through the years, but these characters are in some cases insufficient to classify related species, and identification of plants from dry specimens are very much tough. And as some of the natural features gets lost when the specimen gets dried up. And also reproductive characters, more appropriately the floral characters, which are considered to be most reliable identifying feature in plants, are very much limited and unusual in case of the bamboos. In such cases, phytolith characters can serve as an identifying feature in absence of the floral characters. So the future prospect of work that needs to be done in the present study is a detailed study and analysis of the individual silica bodies from the leaf epidermis as well as the calm sheet, and for proper delimitation of bamboos at genus and species level as well. And these can be done by acid digestion of the leaves as well as the calm sheets, and then study and measurement of the individual silica bodies after extraction by the help of centrifugation and all. So these are the references that I have used for preparing this presentation. Thank you.
Okay, thank you, Uniki, for your nice presentation. Yes, that phytolithic liths, the structures of those phytoliths are very much specific to particular species, and it will definitely help in the identification in the field of taxonomy. So, thank you very much for your presentation. Now, I would like to invite Pooja Singh for our present. The title of our talk is Phytochemical Evaluation and Antioxidant Activity of Two Species of Terrace from Darjeeling District of West Bengal. Pooja, are you there? Pooja Singh, are you there? Okay, now I would like to request next speaker, Sudipa Das. Sudipa Das, are you
Yes,
there?
thank you, sir. Yes, sir. Thank you, sir.
Please present your talk. A title
Oh, yes.
is not on epiphytic bryophytes of
Yes. Hello, Monoranjan, Pooja is there. Hello. Hello.
Pooja Singh is
Hello.
there.
Pooja is there. Pooja is there.
Pooja Singh,
Hello. Hello.
but she is not responding. No. Hello, Pooja.
Yes. Yes, sir. Yes, sir.
Okay.
Yes. Hello.
Sudipa Das.
Yes, sir.
You will present after
Okay,
Pooja.
okay, sir.
Okay. Pooja Singh, please present your talk.
Sir, I am present.
Okay. Go ahead.
Good evening to respected chairperson, respected teachers, and fellow research scholars. Myself Pooja Singh. As you can see on the screen, my topic is phytochemical evolution and antioxidant activity of two species of terrace from Darjeeling district, West Bengal. The present study was planned to extract and analysis of various phytochemical compounds from immature and mature fronds of two species of terrace in methanol and aqueous solvents to determine the total phenol and flavonoid content of various extracts.
the two plant samples to evaluate the antioxidant properties of various extracts of the tested plant. Now I would like to give overview of pteridophytes because their pteridophytes are also known as a vascular cryptograms because they are non-seeded plants containing. They are represented by 144 genera and about 1,157 species in India. Excuse me, sir, my slide is visible. Sir?
Yes, yes, visible.
Okay, thank you, sir.
It is changing.
Purifies research of antioxidants, food and medicine because fern played an important role as antimicrobial, anti-inflammatory, anti-tumor and antiviral and anti-carcinogenic agent. They are used in Unani system of medicine from ancient times for the treatment of various human ailments. There are two types, plants are two. First species is Pteris biota, belongs to family Pteridaceae and commonly known as thin leaf breakfern because its anatomy and it is found to grow in various habitats and has been used as a folk medicine for ages. And another species is Pteris semipinnata, known as herb leaf fern, which belongs to the family Pteridaceae. And it was well known as medicinal fern which cured disease like cleaning heat, removing toxin, and it is indicated for acidic hepatitis and external use for traumatic injury, traumatic bleeding, etc. Let's move to next part, collection and preparation of plant material and extract.
Your slides is not visible.
Not visible slide.
Okay, you
No,
continue.
visible now. Visible now. No.
Okay, you continue.
Okay, sir. The plant materials, their immature ferns without sori and mature ferns with sori were collected from different regions of Darjeeling. The plants were identified and authenticated by Pteridology and Pterido Paleobotany Laboratory, University of North Bengal. Now, now move to next part, preparation of plant extract. After crushing the plant materials, uh, collected separately with different solvents, hot water and methanol, then heated for 4 hours in 30 degrees Celsius. An excess solvent was removed by evaporation and make up the final volume 500 mg per ml.
What happened, Puja?
Sir, some technical issues was there.
Okay, then you present later on. Okay. So, Sudeepa Das, are you there?
Okay, sir.
Yes, sir. Yes, sir, I'm here.
Puja, you present later on. Okay. Yes, Sudeepa Das, Bhattacharya, please present here.
Okay, so let me share my screen.
Okay. Please try to complete within time. Okay.
Yes, sir. Sir, is my screen visible?
Not now.
Just a minute, sir.
You minimize that Google Meet screen. Okay, now visible. Please unmute yourself.
Sir, am I audible?
Yes, yes.
Okay. Good evening, everyone. Myself, Dr. Shudipa Dash, Assistant Professor in Botany, Sundarban Haji Desharat College. And today my topic of presentation is a note on epiphytic bryophytes of Southern Assam, India. Now, what are epiphytic bryophytes? Epiphytic bryophytes are fascinating growth forms which are significant components of biodiversity in a forest ecosystem. They comprise at least 10% of a forest vegetation. They grow mainly on barks of stems or twigs, which are known as corticolous, and they can grow on leaves either, known as follicolous or epiphyllous. They mainly grow on spermatophytes like angiosperms or gymnosperms, or even on fern fronds. The host spermatophytes, which are commonly known as sporophytes, they can be stem, twig, or leaves, or even the aerial roots of some host taxa. Now, Southern Assam, being a part of the Indo-Burma biodiversity hotspot, has been identified as an abode of epiphytic bryophytes due to its unique physiographic and ecoclimatic conditions. Now, what are the main conditions which support the growth of epiphytic bryophytes? Number one is the heavy rainfall, extremely moist and shady tropical to subtropical forests. As we know that bryophytes are extremely moisture-loving taxa, and epiphytic bryophytes prefer extremely moist and shady conditions to grow as they are less tolerant to desiccation. They obtain the required water from rain and atmospheric humidity. Next is the foggy and misty weather. As water is the essential part to complete the life cycle, but during winter they don't get the liquid water. So they collect the required moisture from fog and mist present in the weather. As Southern Assam experiences foggy and misty weather in winter, so it has become a reservoir for growth of epiphytic bryophytes. And number three is the waterlogged condition of the forest floor. Southern Assam experiences a high percentage of rainfall during pre-monsoon and post-monsoon, which prevails the flooded condition of the forest floor. And in such condition, the ground-loving taxa, terrestrial taxa, they cannot grow. They eventually die. And this condition has promoted the growth of facultative epiphytes. That means the terrestrial forms, they grow as facultative epiphytes. Now, these are the findings. I have just jotted down some prolific taxa. It has been found that two main groups, that means the liverworts and mosses from the bryophytes, grow as epiphytes. Among the liverworts, the main family which has been identified as epiphytic bryophyte taxa are Legionnaceae. Some main genera are Archilegiunia,
Dr. Bhattacharya, your slides are not moving.
Not moving, sir.
To me,
am I stuck to the first slide?
Please check with the coordinator, sir.
Sir, which slide is visible? Can you tell me?
First slide.
The first slide only.
You just click on the slide.
Okay. Sir, I am clicking on the slides. So is it visible now?
It is visible but not changing.
Okay. Sir, Just a minute. Sir, is it visible now?
No.
Sir, let me share my screen again. Sir, is it visible now? The second slide?
No. Go for full screen. Not
Now,
working.
sir?
Not working.
Sir, I think we should.
Sir, now is it? Sir, now is it visible?
No.
Yes, yes, yes.
Yes, yes.
Sir, I am showing the third slide. Is it visible now?
Yes, yes.
Okay. Sir, I have just discussed this part. Can I move to the next slide? That is
Yes,
the prolific
yes,
taxa.
yes.
Okay. Sir, I have just listed the prolific taxa here, and it has been recorded that mainly the family Legionnaceae from liverworts are the prolific taxa. Namely, some species are Archilegionia, Cordalegionia, Legionia, Leptolegionia, etc. And from mosses, the main species are Entodon, Scariosus, Arpodium, Erythrodontium, Fabronia, and Macrometrium. Now, it has been found that some taxa like Frulania, Entodon, and Arpodium, they have habitat specificity. That means they grow on some specific host. Rest of the taxa are not host specific. They grow on broad-leaved evergreen and deciduous trees. And taxa like Arpodium and Erythrodontium, they have specific requirements for their microenvironmental factors, and their occurrence has been found to indicate microclimatic condition of that particular area. And diversity of epiphytic bryophytes are found to increase with diversity of their host taxa. Genera like Colulegionia and Legionia, they can use wide range of hosts, and thus they form the main part of the epiphytic bryophyte flora. And it has been found that the taxa which grow on deciduous leaves, they form some vegetative propagule-like structures like gemi to complete the life cycle in a more reduced time, as before completion of before shading of their temporary substrate, that is the deciduous leaves. Now, their relation with the hosts, some dominant host taxa are Anthocephalus, Artocarpus, Melina, Nitam, Musa, etc. And it has been found that epiphytic bryophytes, they choose host taxa depending upon the physical as well as the chemical nature of the phorophyte. Mainly, they prefer to grow on rough and corky stem bark, as because the corky nature of the stem, it soaks up water and retains water, which supplies the bryophytes moisture. And it has also been found that the architectural pattern of the stem, like some leaf traces retained on the palm stems, it also hosts the epiphytic bryophytes. As the grooves of these leaf traces, they accumulate nutrients and substrates, humus like structure, like things, which supports the growth of the epiphytic bryophytes. And it has also been found that the acidic nature of the stem, it doesn't support the growth of the epiphytic bryophytes. They don't prefer the acidic nature of the stem, and hence on the Gymnosperm taxanitum, only the leaves have been found to support the growth of the epiphytic taxa, where the stem is not participating as the porophyte due to its acidic nature. And the benefits offering to the hosts, the epiphytic bryophytes are found to have a significant effect on the microenvironment of their host and on the ecosystem where they are abundant. They hold the water canopy and decrease water input to the soil. And the epiphytes create a significantly cooler and moister environment in the host plant canopy, potentially greatly reducing the water loss by the host. They also stop the loss of environmental water and thus play an important role in the hydrological cycle of the nature. And in conclusion, we can say that epiphytic bryophytes are very much vulnerable and they are much prone to altered microclimate. Mainly among them, the folliculus taxa, that means the taxa growing on leaves, they are much more sensitive to their phyllosphere. That means the microenvironment created by their host leaves. And hence, opening up a forest canopy due to deforestation and any change, that means invasion of exotic non-host taxa, any change in their microclimate, moisture, temperature regime, anything that is destructing these tiny vulnerable species. And as these taxa are not very much important economically, so we are paying less attention on them. So this is the high time to think about the proper conservation strategy of these tiny, significant yet neglected plant groups. Thank you. Thank you, sir.
Hello.
Sir,
হ্যাঁ হয়ে
sir,
গেছে?
completed. Yes, sir.
Okay,
Thank
okay.
you.
Thank you, Dr. Das, for your presentation. Thank you. Now I invite Pooja Singh. Are you ready? Okay, Pooja Singh, please present your talk.
Sir, I think we should go forward because we are waiting.
No, she is now doing from my lab, so I think this time she will not face it.
Okay.
Uh,
She's starting.
Please start. And
Sir,
Huh?
please move
Huh?
forward, sir,
Okay.
with the next participant.
Next participant, please. Anwar NP. Please present your talk.
Sir, yes.
Yes, yes, please.
Audible, sir?
Yes, you are
Yes.
audible.
Okay. Oh, ya. Itu ya. Itunya dua. Good evening, everybody, sir. Good evening, sir. Sir, is audible?
Please, you are
Okay.
audible. Please
Okay.
start.
I am going to present my topic entitled as Phytochemical Analysis and HPTLC Profiling of Cryptocorin spiralis variety spiralis and Cryptocorin spiralis variety cogenetoides. Cryptocorin, commonly called water trumpet, is an aquatic aroid to Southeast Asia. Many species are used as aquarium plants, and some other, for example, like Cryptocorin Cryptocorin retrospiralis, Cryptocorin crispatula, and some other species like Cryptocorin spiralis, variety spiralis, Cryptocorin retrospiralis, etc., are used as food supplement and also herbal medicine. These medicinal properties is mainly due to the presence of various bioactive compounds present in these plants, in addition to carbohydrates, lipids, proteins, and calcium oxalate crystals. This plant is characterized by specific inflorescence. In India, it is represented by 7 species, namely Cryptocorin ciliata, Cryptocorin cogenata, Cryptocorin cansobrina, Cryptocorin retrospiralis, Cryptocorin shivadasini, Cryptocorin tambravaraniana, Cryptocorin spiralis variety spiralis, Cryptocorin spiralis variety cogenetoides. The present study is aiming to identify the various phytochemicals present in the rhizomic extracts of Cryptocorin spiralis and Cryptocorin spiralis variety cogenetoides, and to develop HPTLC fingerprint profiling for identifying the specific compounds. Other objectives of my study: to extract and analyze the phytochemicals both qualitatively and quantitatively, to analyze the mineral content of the rhizomes of the two varieties of cryptocorin, then HPTLC profiling of the two varieties of cryptocorin, and also to develop efficient solvent system for HPTLC profiling. Coming to materials and methods. Field study. The two varieties of plants, namely Cryptocorin spiralis variety spiralis, are collected from the paddy field of Malabar region of Kerala, and also another variety, Cryptocorin spiralis variety cogenetoides, is collected from Borbet, Gaganbauda, Maharashtra, Kolhapur district of Maharashtra. Then extraction. Phytochemical extraction is of the dried rhizome is done by using hexane, chloroform, ethanol, and water by the help of oxalate apparatus. And preliminary phytochemical analysis was done by the method proposed by Habon and Cockate method. Then quantitative analysis. In quantitative analysis, determination of total flavonoid content is estimated by Session et al. Method, and the total phenolic content is estimated by Follin Siaklat assay by proposed by Singleton et al. Method. Then mineral analysis. It is done by acid digestion method of Toth et al. Proposed by Toth et al. And then HPTLC profiling. HPTLC profiling of this hexane, chloroform, and methanolic extract were performed by CAMAG HPTLC system method proposed by Haban method. Then statistical analysis is done by using ANOVA version 18. Coming to result and discussion. First of all, Cryptocorin spiralis is a very common spiralis, variety spiralis is a very common species found in paddy field along the banks of rivers, streams, springs, grass plains, swamps, etc. It is Some part of Kerala, some part of in Kerala, the leaves are used as food supplement, and it is used as a substitute for aconitum metrophyllum to treat diarrhea, and also used to against cough, fever, nausea, abdominal pain, and vomiting in infants. Cryptogoryn spiralis variety cognitoides is endemic to Maharashtra, mainly found in Sindhudurg, Ratnagiri, and Kolhapur district. This is a photograph of the two plants. Then preliminary phytochemical screening. Preliminary phytochemical screening showed the presence of phytochemicals like carbohydrate proteins, alkaloids, glycosides, sterols, phenolics, flavonoids, fixed oils, and saponins. And quantitative assay. Quantitative assay total phenolic content is higher in Cryptogoryn spiralis variety cognitoides, around 4.1 plus or minus 0. Okay. In Cryptogoryn spiralis variety spiralis, it is about 3.7 plus or minus 0. And the total flavonoid content, it is about 0.3933 plus or minus 0.012605 milligram per quercetin equivalent per 100 gram in spiralis variety cognitoides, and 0.3133 plus or minus 0.01699 in Cryptogoryn spiralis variety spiralis. And next, the mineral analysis. Mineral analysis of both the plant shows adequate amount of nitrogen, phosphorus, potassium, sodium, manganese, zinc, copper, iron, and boron. The amount is high in Cryptogoryn spiralis variety cognitoides than the Cryptogoryn spiralis variety spiralis. Specialty profiling. Specialty profiling is done by using three solvents: methanolic extract, chloroform extract, and hexane extract. Specialty fingerprint of profiling of Cryptogoryn spiralis variety cognitoides. We develop a toluene ethyl acetate methanol 7:3:1 ratio for methanolic extract, and it get about 10 peaks with different RF values. In the same plan with the chloroform, we used toluene ethyl acetate in 8:2 ratio, and contain six number of peaks with the 6 different compounds. The same plan with hexane, we used toluene ethyl acetate in 9:1 ratio, and possess 13 peaks with 13 different compounds. Then specialty profiling of Cryptogoryn spiralis variety spiralis. Here also we are using three solvents: methanol, chloroform, and hexane with the same ratio. Methanolic extract possess 9 peaks with 9 different compounds. Chloroform with 4 peaks with 4 different compounds, and hexane with 8 peaks with 8 different compounds. This is the densitogram of methanolic extract to 254 nanometer. Next, visible light. It's a fingerprint of methanolic extract for plants. The densitogram of chloroform of extract at 254 nanometer at 366 nanometer at visible light and fingerprint. Densitogram of hexane extract at 254 nanometer, 366 nanometer, visible light, and also the fingerprint. In the present study, I conclude that total phenols and flavonoid content is higher in Cryptogoryn spiralis variety cognitoides than the Cryptogoryn spiralis variety spiralis. And the best solvent system for methanolic extract is toluene ethyl acetate and methanolin 7:3:1 ratio. And for chloroform extract, toluene ethyl acetate in 8:2 ratio, and hexane extract in toluene is to ethyl acetate in 9:1 ratio. Herewith I conclude, sir.
Okay. Thank you, Anwar NP, for your talk on that phytochemical analysis, HPLC profiling of two species of Cryptogoryn for southern part of India. Thank you very much for your presentation.
Welcome, sir.
Now, I would like to invite Dr. Manojit Devnath, to present his topic on
Yes, sir.
cyanobacterial morphotypes from terrestrial biofilms of lower Gangetic plains of West Bengal.
Thank you, sir. Good evening to all the participants and the respected chairperson of this session. I think I am the last speaker, almost the last speaker of this session. So I will not bore you, but I will start my presentation. So in this auspicious occasion of this the national seminar, the diversity of food and the nutritional security, I would like to present cyanobacterial morphotypes from terrestrial biofilms of the lower Gangetic Plain, West Bengal, India. You can think why I am choosing this one regarding the food diversity and nutritional security. Because from the morning in the inauguration session, our chief conservator of forest also addressed various type of plants in forest along with algae and cyanobacteria and their role regarding the soil fertility. So first I will say few words about biological crust and biofilms. What is biofilms? Biofilms are the film or coating or layer where many organism or few organism can board within a mucilaginous substratum, and this is called the biofilm. And when this biofilm gradually become matured, it will creating the biological crust. That crust may be on the soil, that crust may be on the rock, that crust may be on the any other substratum. And I am focusing here on the edaphic crust. What are their role? These crusts are essential for aggregation of mineral particle at the soil surface, and by that way they are gradually extend their role for the stabilization of soil, fixation of nitrogen, carbon, regulation of water cycling, or in one term, the biogeochemical cycling of the soil. And by that way, they are offering manifold approach for the vascular plants, which are the key source of our food as crop or any other fruits. In case of natural environment, they are reducing seedling, they are enhanced or reducing seedling establishment, creating the natural forest system, promote the chemical and physical weathering, and provide wildlife habitat along with the regulation of soil food web interaction. Therefore, keeping this view in our mind, the study of the present, the focus of the present study is threefold. The survey of cyanobacteria from two distinct habitat, for example, edaphic crust, one I have deleted. Another one, the understanding the role of environment gradients on the cyanobacterial occurrence, because they are the pioneer colonizer and the keystone species in some environment or microhabitat, and along with that, they are creating the space for other primary producers. And the case studies on taxonomic viewpoints as revealed from the surveyed area with cyanobacterial morphotype of interests. Therefore, with a long span, I have collected very wide range of samples from Sagar Island to the Sandakphu. As we are saying that these are sample survey, because West Bengal is representing the 7 biogeographic distinct ecoregion, and along with that there are coastal estuarine soil, alluvial plain, lateritic zone. Along with that, Himalayan high mountain habitats, including terraituas. So workflow, field survey, environmental parameter and in situ laboratory study, collection of naturally occurring substratum with cyanobacterial visible growth, reweighting them, isolation on agar plate, enrichment. Then, with the aid of microscopy like stereo zoom, light microscope, confocal microscope, and field emission scanning electron microscope, we can explore the thallus texture and morphological details for taxonomic study. And this taxonomic enumeration follows the monographs and published literature for identification of species. And along with that, species environment and sampling site correlation analysis is very much important. And finally, molecular phylogeny will help us to predict whether the species or the diversity is present.
Dr. Devnath, your slide is not moving.
Not moving, sir?
Uh,
Now, sir?
On which slide you want to?
I am, sir, on workflow.
Workflow. No. You are on first slide.
Okay, sir. Now, sir?
Yes. Now, okay.
Okay, then I will not do the full screen. Okay.
No problem. You go ahead.
Okay. So, as I said in workflow, if we follow the things, I have collected one edaphic crust which from Sandakphu on rock. Edaphic crust on rock. This is not lithophytic crust. Okay. As I put the knife. And when I am going to the fluorescence microscopy, in the sample in the photograph C, almost nothing is visible except one or two red dot, because very few are alive in this dot condition. And when I am going for FESEM study, what I have derived that there are filamentous cyanobacteria or the filamentous organisms are present after reweighting. What I am getting in fluorescence, the red auto-emitting chlorophyll A fluorescence with filaments and other masses. And when I am going to the light microscope again, to the right hand side photographs, two photographs in green color, I am seeing that filaments and unicellular coccoidal one. So these are the scenario and the workflow we can follow for our study. Now coming to the edaphic cast, which were collected from various soil habitat or edaphic habitat, as I said earlier, these are the list. And this is the CCA redundancy analysis, that is species environment correlation analysis along with the collection sites. That means what kind of organisms or cyanobacterial organisms are present along with the gradient of which environmental parameters. There are two clear views. One, that is green mark, the mountain, low altitude or high altitude terra forest areas. They are governed by the available nitrogen in the soil, and available iron is the key governing factor for cyanobacterial occurrence in soil crust or edaphic crust in lateritic zone, and organic carbon salinity and available phosphorus all together creating a force for the coastal saline and alluvial cultivated and non-cultivated edaphic habitats. So this is clearly visible. I am not going to the detailed cyanobacterial species because time is short. So what I am getting or the take home message from this study, I have studied that or getting the result like the successional stage in the cyanobacterial community on edaphic crust. What is that? First, Aphanothece glyocapsa, the unicellular colonial one. Later, the filamentous, simple filamentous one, that is with motion and non-motive, that is Leptolimba and Oscillatoria. Third is the Microcoleus and Nostoc. They are the keystone one because they are producing huge mucilage and spreading hugely with their filamentous movement to the soil surface. And fourth, these are almost stabilized soil crust with Lyngbia and Cytonema. Those are producing very hard structural sheath which are bearing the cytonemin and microsporin like amino acids or protecting the other layers from the UV radiation and high sunlights and also from desiccation too. And lastly, we are finding the Westeliopsis fissurella group in case of especially the cultivated and old soil part of the alluvium zone. Now I am coming to the searching of new species from nature. How we will do that or how we will proceed. In that case, I am representing 3 species for case study. One from Kolani, non-cultivated alluvial soil. Another from Karimpur, from the district of Nadia, West Bengal. Cultivated alluvial soil, and that is affected by very high arsenic content due to the irrigation water. And Sagar Island as estuarine mangrove soil. And the composition of these 3 type of soil is given here. Regarding the cyanobacterial species richness means by the numbers. The lowest in the arsenic affected, highest in the Sagar Island estuarine mangrove area, and medium in the non-cultivated alluvial soil. So this is the 2 morphotypes of interest. One is Gatlerinema CF calcutense and another is Gatlerinema CF jassurvense. They are almost similar, but their tips are different. One is acuminate, another is blunt, round, conical. These are the FESM photographs of these 2 groups. And now I am coming a species nova which we published as Leptolyngbia indica. And these are the comparative table with the Leptolyngbia database throughout the globe, and where our Leptolyngbia indica situated in the 8th serial as their substratum and subobtuse to obtuse end cell structure. This is very important regarding the Gatlerinema or Leptolyngbia. This is the overall morphotaxonomic study, the light microscopy, the scanning electron microscopy, the confocal microscopy, along with the 16S rRNA phylogeny analysis. Here we can see the red color underlined part in the bootstrap value. It is very unique along with the other Leptolyngbia species existing from the NCBI data bank and the Japan data bank, along with the distinct clade representing the Leptolyngbia in the Leptolyngbia and other phylogenetic ML tree with distinct bootstrap value too. Another is the Coleofasciculus, which was earlier known as Microcoleas. Why I am putting CF likely to be? Because they are not studied by molecular phylogeny, because in recent trend the polyphasic approach along with ecology and molecular phylogeny we can use the final species identification in case of cyanobacteria. So this is the Coleofasciculus. Another one, it is very important one. Basically this is from crop fields. When waterlogging condition, you can find that green to brownish color mat, and this
Dr. Devnath,
like
try to wind up, please.
Yes, sir. So this is very rich in phycoerythrin. And what is our take-home message? We should not neglect the community on soil surface and do not eradicate soil crust on bare and cultivated land surface until unless we very much require to that. And the development of interest and knowledge on autotrophic biofilms and trust among the students and researchers, explore the diversity on various ecological regime regarding the biocast, and establishment of culture in laboratory for further exploration of their functionality for environmental remediation, agriculture, and natural soil health. So thank you for your patience. And this the part of the research partly in along the stretch is funded by ACRB, the West Bengal Biodiversity Board, Department of Biotechnology, Government of West Bengal, and CSIR, Government of India too. Thank you to all.
Okay.
So I have finished.
Thank you very much, Dr. Monojit Devnath, for your such a nice findings, new species of algae from edaphic situations from various climatic zone of India.
Thank you,
Congratulations
sir.
and best of luck for your future research. So, dear participants, in this session, we have two lead lecture, one by Dr. Gyan Vikas Bhandari, another one by Ram Sarup Meena from Benares Hindu University. And in total of 15, one person, that is Pooja Singh, cannot be present due to some network issue. So in total 15 presentation were there, and all the presentation interestingly covering all the classical part. That is the group, that is the algae, then fungus, then bryophyte, pteridophyte, gymnosperm, angiosperm, all groups. So it was a very nice session, and along with the phytochemical analysis and the phytolith analysis, endemic species, biodiversity. So everything is included within this session, and I hope it is a very important that the identification or the description of new species is very important. Finding of new economically important species through traditional knowledge is very important issue, and that all are related to the nutritional food diversity, and that is that issues of diversity and food safety and security. So thank you very much to all the participants for your nice, such informative findings that you have shared with us in this 2 days national seminar. We are very much thankful to you all
Excuse me,
and the
sir.
uh,
Sorry, sir. Sorry to interrupt you, but I'm still in the queue, so I'm waiting my name to be announced.
sorry, your name
I
is
am Prasinna Rai. I am Prasinna Rai. I have already.
Piyush, the list, her name is not included.
So yes,
So she
yes,
will present tomorrow, no problem.
sir. So actually, I have a request. Tomorrow I'll be traveling. So, is it possible? So I'll be very quick.
Okay, then go ahead. Present
O-
your seminar.
Okay, sir.
শুনে না ওরটা কি বলছে হয়ে যাক
So is it visible?
yeah visible.
Oh, I'm sorry. What I've done. Good evening, respected dignitaries, respected teachers, and dear participants. Myself, Prasinna Rai, and today I'm going to talk about diversity and species composition of vascular epiphytes in subtropical forest of Darjeeling, Himalaya, India. One of the most conspicuous component of our ecosystem is vascular epiphytes. Epiphytes are the plants that attach themselves to and grow on the. Sir, am I audible?
Yes, you are
You are
audible.
audible. Okay,
Sorry, sir.
go
Okay.
ahead.
The epiphytes are the plants that attach themselves to and grow on the other plant, but in contrast with the parasitic plants, they do not extract nutrient from the host plants. The host only provides a place to grow. Some of the significance of vascular epiphytes are they contribute substantially to ecosystem diversity, production, and nutrient cycles. They also act as global indicator for climate change. They have contributed immensely to the forest biodiversity, and apart from that, they have provided food and shelter for fauna. And some epiphytes have also found have also ethnomedicinal uses. The main objectives of the present study is to study the diversity and species composition of vascular epiphytes and to understand the association of vascular epiphytes with different host traits. The study was conducted in the subtropical forest of Darjeeling, Himalaya. The table in the slide shows the different vegetation types of Darjeeling, Himalaya. So for the epiphytic sampling, several field surveys were conducted during the pre-monsoon, post-monsoon, and during the winters. The voucher specimens uh, were collected and identified, and the further studies were made. Properly maintained herbarium exegesis has been maintained for future study. So, a total of 55 species under 39 genera belonging to 17 families was observed. Orchidaceae was the most dominant and abundant family, followed by Polypodiaceae and Araceae. Majority of the species recorded in the study area were herbaceous and perennial in nature. Here, figure 1 shows the families with number of species and genera. Similarly, figure 2 shows the percentage distributions of taxa, like orchids were represented by 40%, followed by other angiosperm, that is 36%, and ferns were represented by 24%. These are the table of distribution of taxa from the study area. Again, the recorded specimens were divided into three categories, such as monocots, dicots, and ferns. Monocots were represented by 23% of the families, and 59% of the families were represented by dicots, and ferns were represented by 18% of total family. Apocynaceae was most dominant among dicots, and Orchidaceae and Polydaceae were most abundant monocot and fern families, respectively. These are the photographs of some of the species that I have recorded during my field service. So phenological status of flowering plants was also observed. Maximum species, that is 43%, bloomed during spring season, followed by 29% that flowered in summer season. Only 21% and 7% flowered during autumn and winter season, respectively. So the distribution within the host tree, the distribution of the epiphytes within the host species was also investigated with the most dominant and favorable host trees. Our results reveals that the number of the species significantly decreases from trunk zone to the inner crown zone. To study the correlation of epiphytic diversity with different host tree traits, dominating host trees were identified during field surveys. CBH greater than 15 cm measured, and the bark texture were also recorded. Bark samples were collected to estimate the pH. We plotted the scattered plot were plotted against the epiphytic diversity and CBH, bark texture, and epiphytic diversity and pH of the host bark, and it was observed that CBH was significant and positively correlated to epiphytic diversity. Host tree having rough bark had maximum species. No significant relations between the bark pH with the epiphytic diversity was found. I would like to conclude here. The present study demonstrated the diversity and composition of vascular epiphytic plant in subtropical forest of Darjeeling, Himalaya, and this study would definitely provide significant information for regional flora as well as flora of Eastern Himalaya hotspot. However, it has been observed that deforestation, collection of plants for commercial purposes, constructions, and agriculture practices prevailing in the study area have been causing frequent change in species diversity and composition of vascular epiphytes and other taxa as well. Many of the taxa are under low population count, therefore there lies a role of conservationist, taxonomist, ecologists in collaborations with forest departments and concerned participation of the locals for proper conservation of the rich and valuable epiphytic taxa in the region. Although the relationship between epiphytic diversity and host characters are studied well, very little is known, thus the host epiphytes association should be studied in future at a greater scale. I convey my sincere gratitude to my PhD supervisor, Dr. Saurabh Mokhtan. Without his kind direction and proper guidance, this study would have been a little success. I am thankful to all my teachers and fellow lab mates, Department of Botany, Calcutta University. I would also like to thank UGC for financial assistance and Forest Department for all the necessary permissions. Last but not the least, I would like to thank all entire organizing committee, North Bengal University, for giving us this and for giving this auspicious opportunity. Thank you so much.
Okay. Thank you, Prasinna,
Thank you,
for your
sir.
presentation. Thank you very much. Okay. So in this session, in total 16 OP. Huh? So over to you, Tornendu. Now I
Okay.
think you can
Sir, I
close the
think it was mistakenly my name
Uh,
has gone to poster presentation, I think.
no,
Okay.
Prashina's presentation was in technical session 2. I think you have wrongly entered into the different link. And that's why
Oh,
there was a
Okay.
oh, I'm sorry,
mistake.
sir, for the
No
inconvenience.
problem. You have already presented. So
Okay,
there is
thanks.
no problem.
Thank you, sir.
So, so we have come to the end of this session. So I would like to thank the session chair who have done a tremendous work. Our own head of the department, Dr. Manoranjan Chaudhary, associate professor. So it was a very extensive session with so many lectures, and all of you have presented very nicely. Though there were some problems with the internet connectivity and all that we cannot control. So that is a part and parcel of the this presentation. So we have come to the end of this session. So we will officially end this session today. And I request all the participants to join tomorrow also for the different plenary sessions and the technical session. So we anticipate a active participation from all of you tomorrow also. So we will be sharing the Google Meet link with all of you also tomorrow and the YouTube links also. So kindly join with us. Thank you.
Okay. Thank you all. Thank you.